So here's something that might surprise you. Right now, as you lie there, something like 86 billion cells inside your head are exchanging tiny electrical pulses, thousands of times a second, and the pattern of those pulses is, in some way nobody fully understands yet, the sound of my voice arriving in your mind. You are not just hearing these words. You are building them, cell by cell, in the dark, right now, without doing anything at all. Tonight we're going to go through all of the human brain, from the very beginning, the history of how people first tried to understand it, the core ideas that took centuries to work out, all the way through to how it actually works today, down at the level of a single cell, talking to another single cell. By the end, you'll understand what a thought actually is, physically, not as a metaphor, as a real thing, made of real parts, that you can picture. If you fall asleep somewhere along the way, that's perfectly fine. That's the point. There's no test at the end of this, no reason to fight to stay awake. The ideas will still be here if you want to come back to them, and your brain, this very organ we're talking about, will keep working on what you heard even while you sleep. That's actually part of how it learns. So let go. Whenever you're ready, we'll start at the beginning. The beginning is stranger than you'd expect, because for most of human history, almost nobody thought the brain mattered at all. Go back about 3,500 years, to Egypt, to the banks of the Nile, to the workshops where bodies were prepared for the afterlife. The embalmers who mummified the dead were, in a very real sense, the first people in recorded history to handle a human brain directly, over and over, thousands of times, across generations. And here is the strange part: they threw it away. When a body was prepared for mummification, the heart was left in place, wrapped carefully, considered essential, because the heart was believed to be the seat of thought, memory, and feeling. The brain, meanwhile, was removed through the nose with a hooked instrument, broken into pieces and discarded, or sometimes just left to liquefy and drained out. It was considered packing material, stuffing, not important enough to preserve. We know this because of a document called the Edwin Smith Papyrus, named after the American who bought it in Luxor in 1862, though the writing itself is far older, likely copied from a text written around 1700 BCE, which itself may have drawn on knowledge even older than that. It's a surgical text, describing 48 case studies of injuries, mostly to the head, and it is, remarkably, the oldest known writing that uses the word we would now translate as brain. The physician who wrote it, whoever they were, describes a patient with a head wound so severe that the surface of the brain is visible, and they note something that must have been startling to observe. They describe the brain pulsing, moving with each heartbeat, corrugated like the folds you'd find inside molten copper as it cools. They noticed that when this particular kind of head injury occurred, the patient sometimes lost function on the opposite side of the body. A wound on the left side of the head, and paralysis showing up on the right side of the body. This is, as far as we can tell, the first written observation of what we would now call contralateral function, the fact that the two sides of the brain each control the opposite side of the body. And yet, even having observed this, even having written it down, the author of the papyrus does not conclude that the brain is the origin of movement or sensation. The observation sits there, accurate, unexplained, disconnected from any larger theory. They saw the wiring working. They just didn't know it was wiring. This tells you something important about how understanding develops. Observation almost always comes before explanation, sometimes by centuries. People can watch a true thing happen, write it down accurately, and still have no framework for what it means. Move forward almost 1,000 years, to Greece, to a city called Croton, in what is now southern Italy, home to a thinker named Alcmaeon, who lived around 500 BCE. Alcmaeon is one of the first people we have any record of who dissected animals for the purpose of understanding how bodies work, rather than for religious ritual or food. He is credited with tracing the optic nerve, discovering the pathway connecting the eye to the brain, and from this and other observations, he proposed something radical for his time. He proposed that the brain, not the heart, was the seat of understanding, sensation, and thought. He believed the brain was where perceptions were gathered together and turned into memory, and where memory turned into experience, and where experience turned into wisdom. This was not the majority view. It was one voice among many, and for a long time it stayed a minority position. The dominant view, held by many respected thinkers of the ancient world, was what we now call cardiocentrism, the heart-centered view. And its most influential champion was Aristotle, writing around 350 BCE, whose authority on almost every subject was so total that his views on this one carried forward for well over 1,000 years after his death. Aristotle had actually dissected animals himself, extensively, and he knew the brain existed and had a particular structure, but his conclusion was that the heart was the control center of the body, the source of heat, sensation, movement, and thought, the first organ to form in an embryo and the last to stop at death. The brain, in Aristotle's system, served mainly as a kind of radiator. Its job, he believed, was to cool the blood that the heart heated up, moderating the body's temperature so the heart didn't overheat itself with its own passionate activity. Aristotle noticed that the brain was cool and bloodless to the touch compared to other organs, and that seemed to fit the cooling theory rather well. He also noted, correctly, that the brain has no sensation of its own when touched directly, which he took as further evidence that it was not the seat of perception. Why would the organ of feeling be the one part of the body that couldn't feel anything? It's worth sitting with how reasonable this all sounds, because that's the real lesson buried in this history. Aristotle was not careless. He was one of the most careful observers in the ancient world. He looked at real evidence, the coolness of brain tissue, its lack of sensitivity to touch, the heart's obvious connection to strong emotion, the way your chest tightens with fear, the way your heart pounds with excitement, and he built a coherent, defensible theory from what he had. He was wrong, but not carelessly wrong. He was wrong in the way that careful, evidence-based thinking can still be wrong, when the tools available aren't yet good enough to see the deeper mechanism. This will happen again and again in this story. Being systematic is not the same as being right, and both things are worth holding in mind as we go, that early ideas, even mistaken ones, were built by serious people doing serious work, and that even serious work needs revision as tools improve. While Aristotle's cardiocentric view was taking hold in the broader philosophical world, a very different tradition was developing in medicine, in the school of thought associated with Hippocrates of Kos, who lived slightly before Aristotle in the late 400s BCE. A text from this tradition, called On the Sacred Disease, addresses epilepsy, a condition that at the time was widely believed to be a curse or a punishment from the gods, a divine affliction, which is where the name sacred disease comes from. The author of this text, writing in the voice of Hippocratic medicine, makes an argument that reads as startlingly modern. The text states, in essence, that epilepsy is no more divine than any other disease, that it has a natural cause just like everything else, and that people only call it sacred because they don't understand it. And then it goes further, making a direct claim about the brain that stands as one of the clearest statements in the ancient world of what we would now call a neuroscientific view of the mind. The text asserts that it is the brain, and only the brain, that is responsible for our joys, our delights, our laughter, our sorrows, our grief, and our tears. Through the brain, it says, we think, we understand, we see, we hear, and we know what is shameful, what is good, what is pleasant, and what is unpleasant. It is the brain that makes us confused or troubled, that fills us with fear and worry, that disturbs our sleep, that causes forgetfulness. All of this, the author argues, comes from one organ, and that organ is the brain. This is, in a real sense, the birth certificate of the idea that your mind is your brain, written roughly 2,400 years before anyone could test it directly. It's a hypothesis, stated with real confidence, resting on clinical observation of patients rather than dissection, since the author had likely observed many people with head injuries and epilepsy and noticed the pattern connecting head trauma to changes in personality, memory, and behavior. But it was a hypothesis competing against Aristotle's enormous philosophical authority, and for a long time Aristotle's version was the one that stuck in the wider culture. The next major figure in this story is Galen, a physician working in the Roman Empire in the 2nd century of the current era, roughly 500 years after Hippocrates and Aristotle. Galen is one of the most important figures in the entire history of medicine, and his influence on how the brain was understood would last, almost unchallenged, for nearly 1400 years. Galen had an advantage the earlier thinkers lacked: hands-on access to a huge number of injured bodies. He worked for a time as a physician to gladiators in Pergamon, treating wounds from combat, which meant he saw an enormous volume of real trauma to the head, the spine, and the nervous system, and he could observe, directly and repeatedly, what happened to a person's body when specific structures were damaged. He also dissected animals extensively, particularly Barbary macaques and pigs, since dissection of human bodies was largely forbidden in Roman culture at the time, a restriction that would shape and limit anatomical knowledge for centuries. Through this work, Galen made a series of genuinely important discoveries. He demonstrated, through direct experiment, that cutting a pig's spinal cord at different levels produced different effects on the body below the cut, showing clearly that the spinal cord was carrying distinct signals to distinct regions, an early and concrete demonstration that the nervous system worked through some kind of pathway, not through some vague general animating force. He also showed that damaging the recurrent laryngeal nerve, a nerve that runs from the brain down into the chest and back up to the throat, caused an animal to lose its voice, a striking and very public demonstration, since he performed this in front of audiences, that a nerve controlled by the brain, running an oddly indirect path through the body, governed something as specific as the ability to make sound. Based on this and other evidence, Galen broke decisively with Aristotle. He concluded that the brain, not the heart, was the source of sensation and voluntary movement, and that nerves carried out its instructions to the rest of the body. This put him closer to the Hippocratic view than the Aristotelian one, and given Galen's enormous authority in later medicine, this helped tip the long-running argument in the brain's favor, at least among physicians, even if it took much longer to settle in wider culture. But Galen also introduced an idea that would prove to be a very long-lasting mistake. He proposed that the brain worked through fluids, specifically through something he called psychic pneuma, a refined vapor or spirit that was created in the brain's hollow internal chambers, the ventricles, and then distributed through the nerves, which Galen believed were hollow tubes, to animate the rest of the body. In this model, thought and sensation and movement were essentially a plumbing system. The ventricles, those fluid-filled spaces inside the brain, were considered the actual site of mental function, not the solid tissue of the brain itself. The soft, fleshy substance of the brain was thought to be secondary, almost a container for the important part, which was the fluid and the empty spaces where that fluid moved and mixed. This ventricular theory of brain function would dominate medical thinking for roughly the next 1400 years, all the way into the European Renaissance, taught in universities, illustrated in medical manuscripts, treated as settled fact. It's a striking example of how a genuinely important correct insight that the brain controls the body through the nervous system got bundled together with a completely incorrect mechanism, fluid moving through hollow chambers and hollow nerves, and the whole package got treated as truth for over 1,000 years, because nobody had the tools to look closer and see that nerves are not hollow tubes at all, and that the solid tissue, not the empty ventricles, is where the real activity happens. Between Galen's era and the European Renaissance, roughly 1,000 years apart, it would be a mistake to imagine that no serious work on the brain and nervous system was happening anywhere in the world. During this long stretch, often thought of in a purely European frame as a quiet period, scholars working across the Islamic world, in centers of learning stretching from Baghdad to Cairo to Cordoba, were actively translating, preserving, questioning, and in a number of important cases directly correcting Galen's original work, keeping this body of knowledge alive and continuing to refine it during a period when access to it had considerably narrowed in much of Western Europe. The physician Ibn Sina, known in Europe by the Latinized name Avicenna, working in the early 1000s, produced an enormous and enormously influential medical encyclopedia, the Canon of Medicine, which organized and systematized existing knowledge of anatomy and disease, including detailed discussion of the brain and nervous system, and which would go on to serve as a standard medical textbook in European universities for several centuries afterward, translated into Latin and used well into the 1600s in some institutions. Ibn Sina also engaged directly and critically with Galen's ventricular theory of brain function, working to map specific mental faculties, including common sense, imagination, and memory, onto specific ventricular chambers, a scheme that, while we now know it was built on Galen's mistaken premise about the ventricles being the true seat of function, nonetheless represented a serious, careful attempt to localize distinct mental functions to distinct physical structures within the brain, an early ancestor of the same basic localization approach that would later produce real, lasting insight in the hands of Broca, Wernicke, and Penfield centuries afterward. The physician Ibn al-Nafis, working in Cairo in the 1200s, is best known today for correctly describing pulmonary circulation, the pathway blood takes through the lungs, several centuries before this was independently rediscovered in Europe, directly contradicting one of Galen's own anatomical claims in the process, and his willingness to challenge specific, well-established Galenic claims on the basis of his own careful reasoning and observation reflects the same broader spirit of critical engagement with inherited authority that would later characterize Vesalius's work in Europe, even though the two men worked centuries apart and in entirely separate intellectual traditions. And the surgeon al-Zahrawi, working in Cordoba in the 900s and early 1000s, produced a detailed and highly influential surgical text describing procedures and instruments for treating head injuries and skull fractures, including specific techniques for safely managing depressed skull fractures, exactly the kind of severe head trauma the ancient Edwin Smith papyrus had documented, without explanation, thousands of years earlier. Al-Zahrawi's surgical text was translated into Latin and remained a standard reference in European medical training for several hundred years. None of this scholarship, on its own, overturned Galen's ventricular theory or definitively settled the question of what the brain was really made of. That would still require the specific combination of tools and evidence that came together later, in Renaissance Europe and then in Golgi's kitchen laboratory centuries after that. But it's worth being accurate about the real shape of this history, rather than imagining a simple, uninterrupted straight line running directly from ancient Greece to Renaissance Italy. Understanding of the brain advanced, when it advanced at all across this long middle stretch, through a genuinely international, centuries-long relay of careful scholars working in different places, in different languages, each one building on, translating, questioning, and occasionally correcting the work that had come before them, long before any of the specific breakthroughs we're about to turn to next. It took the European Renaissance, and a very particular kind of stubborn, hands-on curiosity, to build directly on this long-inherited foundation and start correcting it further. The central figure here is Andreas Vesalius, a Flemish anatomist working in Padua, Italy, in the 1530s and 1540s. Vesalius did something that seems obvious now, but was genuinely unusual at the time. He insisted on doing his own dissections, with his own hands, rather than reading Galen's descriptions aloud from a raised platform, while an assistant cut open a body far below him, which was the standard teaching method of the era. This mattered enormously, because when Vesalius actually looked, carefully, at human bodies rather than relying on Galen's centuries-old descriptions, which had been based mostly on animal dissection, he began finding places where Galen was simply wrong about human anatomy. In 1543, Vesalius published a massive, beautifully illustrated work called On the Fabric of the Human Body, and in it, he directly and publicly challenged Galen's authority on multiple points, including anatomical details of the brain. Vesalius could find no clear evidence that the ventricles were the special seat of thought that Galen had described, and his illustrations began the slow process of shifting attention from the hollow spaces inside the brain toward the actual solid tissue, the visible folds and structures that we now know matter far more. This was a genuinely bold thing to do, intellectually. Galen's authority had the weight of over 1,000 years of medical tradition behind it, reinforced by universities, by religious institutions, by simple habit. Challenging him wasn't just correcting a fact, it was challenging a whole structure of trust in inherited knowledge. Vesalius faced real criticism from more traditional physicians of his time for his willingness to say, in effect, "I looked myself, and what I saw does not match what we've been told." This willingness to trust direct observation over inherited authority is really the beginning of what we'd now call the scientific approach to the brain. And it's worth noticing that it required, first, someone willing to get their hands genuinely dirty, cutting open real human bodies himself rather than trusting old texts, and second, someone willing to accept the risk of being wrong in public, of saying an ancient authority had made an error. From here, the story moves into the 1600s, and to a philosopher whose ideas about the brain, while wrong in an important way, still shape how people talk about the mind today, sometimes without realizing it. René Descartes, working in the first half of the 17th century, proposed a very influential picture of how mind and body relate to each other. Descartes believed that the mind, the thinking, feeling, conscious part of a person, was fundamentally different in kind from the physical body, made of some other substance entirely, not extended in space the way physical matter is, not subject to the same laws. This is what's called dualism, the idea that mind and matter are two separate kinds of thing. But Descartes still needed to explain how these two totally different substances, the immaterial mind and the physical body, could interact, how a thought could cause an arm to move, how a pinprick could cause a feeling of pain. And his answer involved the brain directly. He proposed that a small structure near the center of the brain, called the pineal gland, was the specific point of contact between the immaterial soul and the physical machinery of the body, the single place where these two different kinds of substance met and influenced each other. Descartes chose the pineal gland partly because, unlike most brain structures, it appeared to be a single, unpaired structure sitting roughly in the middle of the brain, rather than existing in symmetrical left and right copies the way most brain tissue does. And this uniqueness seemed to him to fit the idea of a single point of unity, where the fragmented sensory information from two eyes, two ears, and so on, could come together into one unified conscious experience. We now know Descartes was wrong about the specific mechanism. The pineal gland's actual job, as we understand it today, is to produce a hormone called melatonin, based on cues about light and darkness, helping regulate the sleep and wake cycle, the very cycle you're moving through right now as you listen to this. It has no special role as a bridge between an immaterial soul and physical machinery. But Descartes' broader framework, the idea of a strict division between mind and body, proved to be extraordinarily persistent in Western thought, and you can still hear echoes of it today whenever someone talks about the mind and the brain as though they were two separate things being compared, rather than accepting that the mind simply is what the brain does, an activity of tissue, rather than a separate kind of stuff sitting alongside it. Untangling that assumption, built deep into ordinary language, took centuries of further work, and in some ways is still being untangled today, including in some of what you'll hear about later tonight. So by the 17th century, several key pieces were finally in place, even if nobody could see the full shape of what they were building toward. The brain, not the heart, was increasingly accepted, at least among physicians and anatomists, as the true seat of sensation and thought. The idea that the brain worked through hollow tubes and empty fluid-filled chambers was starting to crack, thanks to careful anatomists actually looking with their own eyes rather than trusting old texts. And a serious question had been opened about how, exactly, a physical organ made of soft tissue could give rise to something as strange and immediate as the feeling of being you, right now, hearing these words in the dark. What was still completely missing, and would remain missing for another 200 years, was any real understanding of what the brain was actually made of, down at the smallest scale, and how its basic building blocks did their work. Everyone so far had been looking at the brain from the outside, or dissecting it in large visible pieces. Nobody yet had any tool capable of seeing what a single piece of brain tissue looked like close up, at the level where the real mechanism was hiding. That gap between knowing the brain mattered and knowing how it actually worked at the smallest scale is the problem that needed solving next, and it's the problem we'll turn to now. There's one more piece of background worth setting in place before we go further, because it explains why the question of whether the brain was made of separate cells was even a live, open question by the mid-1800s in the first place, rather than something already assumed as an obvious starting point. In 1838 and 1839, two German scientists, the botanist Matthias Schleiden and the physiologist Theodor Schwann, working somewhat separately but building directly on each other's findings, proposed what became known as cell theory, the now foundational idea that all living tissue, whether plant or animal, is composed of individual, discrete cells, each one a basic self-contained unit of life. This was, at the time, a genuinely major unifying idea in biology, drawing together observations that had been accumulating gradually since the 1600s, when the English scientist Robert Hooke first coined the very word cell after examining a thin slice of cork under an early microscope and noticing its small, regular, box-like compartments, though what Hooke was actually looking at in that particular case was the empty cell walls left behind by dead plant tissue rather than any living cellular material itself. By the late 1830s, with considerably improved microscopes and more careful, systematic observation across a wide range of tissue types, Schleiden and Schwann were able to argue convincingly that this cellular organization was a truly general, universal feature of essentially all living matter, not merely a peculiarity of a few specific tissue types like cork or plant leaves. But nervous tissue, right from the very beginning of cell theory's acceptance, stood out as a stubborn, persistent exception to this otherwise sweeping and unifying rule. When early researchers looked at nervous tissue under a microscope, it simply didn't show the same kind of clear, individually bounded units that were readily visible in, say, blood, with its neatly separate round cells floating freely in fluid, or in plant tissue, with its distinct compartments. Nervous tissue looked, as we discussed a little earlier, like a dense, continuous, tangled mass, and this observation led many careful, serious researchers of the era to conclude, quite reasonably given what they could actually see, that the nervous system might simply be a genuine exception to Schleiden and Schwann's otherwise universal cell theory, one true continuous structure, standing apart from the discrete cellular organization found everywhere else in the living body. This is worth appreciating because it means the debate we described earlier over whether the brain was one continuous net or a population of discrete cells wasn't simply an isolated technical puzzle confined narrowly to neuroscience. It was a genuine challenge to one of the great unifying ideas in all of 19th century biology, and settling it in Cajal's favor didn't just clarify how the brain works. It also confirmed, for the very last remaining holdout tissue type in the entire body, that cell theory really did apply universally after all, with no true exceptions remaining anywhere in the whole of living nature. By the middle of the 1800s, scientists knew, in broad strokes, that the brain was electrical. This itself had been a hard-won discovery. In the 1780s, an Italian physician named Luigi Galvani had noticed that a dead frog's leg would twitch when touched by two different metals at once, and through careful further experiments, he concluded that animal tissue itself carried something like electricity, a force he called animal electricity, running through nerves and muscles. This was controversial at the time. Galvani's own nephew, Giovanni Aldini, took the demonstration on tour across Europe, applying electrical current to the bodies of executed criminals in front of horrified and fascinated public audiences, making the freshly dead body's face twitch and its arm rise, in displays that blurred the line between serious science and public spectacle, and that, incidentally, helped inspire Mary Shelley's Frankenstein a few decades later. Underneath the showmanship, though, was a real and important discovery. Nerves and muscles respond to electrical stimulation, and something like electrical signaling is involved in how the body moves. But knowing that the nervous system was electrical did not tell anyone what the nervous system was actually made of, physically, at a small scale, or how the electricity was being carried. And this is where the real bottleneck sat for most of the 1800s. The instrument that would eventually break this bottleneck open was the microscope. But early microscopes, even good ones, ran into a serious problem when pointed at brain tissue. Nervous tissue, when sliced thin and placed under a lens, looked like an undifferentiated, pale, tangled mess. Unlike, say, muscle tissue, which showed fairly clear repeating fibers, or blood, which showed distinct round cells floating in fluid, brain tissue under a basic microscope in the mid-1800s looked like a dense, gray, structureless blur. You could tell there was something there, some kind of fine material, but you could not make out individual units within it. This was the central technical problem blocking progress. The tool existed, the microscope, but the material itself refused to reveal its structure through that tool, because everything in it was roughly the same pale color and packed so closely together that individual components were impossible to distinguish from their neighbors. This mattered because a huge scientific question hinged on solving it, a question that came to be known as the neuron doctrine debate, though it wasn't called that yet in the mid-1800s. The question was simple to state and enormously difficult to answer. Is nervous tissue made of individual, separate cells, each with its own boundary, communicating with neighboring cells across some kind of gap? Or is nervous tissue one single, continuous, interconnected web, a fused net of material with no real breaks in it at all, more like a single sprawling structure than a population of distinct individual units? This might sound like a narrow technical question, but the answer would completely determine how anyone could ever think about what a thought, a memory, or a sensation actually was, physically. If the nervous system were one continuous fused net, then information would have to travel through it more like water flowing through a single continuous pipe system, or perhaps more like electricity flowing through one big continuous wire. There would be no individual switching points, no separate units making individual decisions about whether to pass a signal along. But if the nervous system were instead made of separate individual cells, each with clear boundaries, meeting other cells only at specific points of contact, then something completely different became possible. Each cell could, in principle, act as its own little decision point, gathering signals and then choosing whether or not to pass them onward. And the connections between cells, the specific pattern of which cell talked to which other cell, could itself be the thing that stored information, that changed over time, that got stronger or weaker with use. These are two fundamentally different pictures of how a mind could work, and in the mid-1800s, nobody could tell which one was true, because nobody could see clearly enough to check. Most researchers working with what tools they had leaned toward the continuous net picture, sometimes called reticular theory, from the Latin word for net. This wasn't unreasonable, given what they could actually see. Under the microscopes of the time, brain tissue often did look like one big tangled continuous mass, without obvious gaps. Prominent anatomists of the era, working with real diligence, traced what looked like fibers merging directly into other fibers, with no visible interruption, and built increasingly detailed maps of the brain on the assumption that it was one giant, richly connected, continuous structure, somewhat like a spider's web woven so densely that it had become a single unbroken sheet, rather than the way we now understand the brain to actually work, as an enormous population of separate individual cells. The reason this remained unresolved for so long comes back, again, to the limits of the tools available. A microscope by itself, even a good one, magnifying tissue that has simply been sliced thin and placed on a glass slide, will show you a jumble of translucent, pale gray fibers pressed together, indistinguishable from one another, because nervous tissue does not naturally have any color or contrast built into it that would let a human eye separate one cell's boundary from the boundary of the cell right beside it. What was needed was not just a better microscope. It was a way to make individual cells visible against their neighbors, some kind of chemical process that would selectively color in a small number of cells within a slice of tissue, while leaving the vast majority of surrounding cells completely untouched and invisible, so that the handful of colored cells would stand out clearly, isolated against a blank background, and their full shape, Their branches, their extensions, could finally be traced by eye. This is worth pausing on, because it's a pattern that shows up again and again across the history of science, not just in neuroscience. Sometimes the barrier to understanding is not a lack of raw thinking power or clever theorizing. Sometimes brilliant people, thinking very hard, simply cannot get past a wall built by the limits of their instruments. The question of whether the brain was one connected net or many separate cells could not be settled by philosophy or careful argument alone, no matter how rigorous. It could only be settled by finding some kind of technique that let a human eye actually see the individual boundary of a single nerve cell for the first time in history. And this need, a purely practical, technical need, for a stain that would color in some cells and leave others untouched, is what set the stage for one of the strangest and most important accidents in the history of science. A discovery made not by someone trying to solve the neuron question directly, but by an Italian physician working almost alone, at night, in a small kitchen laboratory, trying something that, by his own later account, he wasn't even sure would work. There's something worth noticing in this too about how real understanding builds. Nobody sets out to invent the neuron doctrine. Nobody wakes up and decides, today I will discover that the brain is made of individual cells. Instead, someone runs into a specific, narrow, practical problem. In this case, a lack of contrast under the microscope, and works for years trying different chemical combinations on scraps of tissue, mostly out of stubbornness and curiosity, without any guarantee that any of it will lead anywhere useful at all. The breakthrough, when it eventually comes, often looks obvious in hindsight, but getting there involves an enormous amount of trial, error, and simple persistence, tinkering in the dark, quite literally, with no way to know in advance which combination of chemicals will finally make the invisible visible. And that's exactly where the story goes next, into a kitchen in Italy in the late 1800s, where an idea would be found that changed everything about how anyone could look at the brain. Before we get to that kitchen, it's worth pausing on a parallel thread of research that was unfolding at roughly the same time, because it answers a slightly different but closely related question, not what the nervous system is made of, but what, precisely, an electrical signal traveling through it actually consists of, at the level of physics and chemistry. This work grew directly out of Galvani's animal electricity experiments from the late 1700s, and it was carried forward through the middle of the 1800s by a German physiologist named Emil Dubois-Reymond, working in Berlin. Dubois-Reymond built increasingly sensitive instruments, including a device called a galvanometer, capable of detecting extremely small electrical currents, and using this equipment, he was able to show, with considerably more precision than Galvani's original crude demonstrations had allowed, that a genuine, measurable electrical change does travel along a nerve fiber whenever it's stimulated, a change he termed the negative variation, since it involved a brief dip in the electrical potential he could measure between 2 points along the resting nerve. Dubois-Reymond spent decades refining this kind of measurement, and he's often credited as one of the founders of the field that would later be called electrophysiology, the careful, quantitative study of the electrical behavior of living tissue. The next real leap came from a physiologist named Julius Bernstein, working in Germany in the final decades of the 1800s, who took Dubois-Reymond's careful electrical measurements and asked a deeper question: what physical mechanism inside the nerve fiber could actually account for this traveling electrical change? Bernstein proposed what came to be called the membrane theory, suggesting that the outer membrane surrounding a nerve fiber was selectively permeable, meaning it allowed some charged particles to cross more easily than others, and that this selective permeability was responsible for maintaining a resting voltage difference between the inside and outside of the fiber, a voltage that then briefly and locally reversed whenever the fiber was stimulated. Exactly the kind of resting potential and brief local reversal we described in detail earlier tonight when we walked through the action potential itself. Bernstein published this theory in 1902, and while a number of the finer details he originally proposed would later need correction, once better tools became available in the 20th century, particularly Hodgkin and Huxley's later, far more precise work with the squid giant axon, the basic core of his idea that a selectively permeable membrane maintains an electrical voltage across itself, and that changes in that permeability produce the traveling nerve signal, turned out to be essentially correct, and it remains the conceptual foundation of everything modern neuroscience understands about how a single neuron generates and conducts its electrical signal. So by the very end of the 1800s, two separate, largely independent lines of investigation were converging on the same organ from two completely different directions, without yet being fully joined together into one unified picture. One line, running through Dubois, Raymond, and Bernstein, was working out the physics and chemistry of the electrical signal itself, the mechanism by which a single nerve fiber generates and carries its pulse. The other line, the one we're about to return to, running through Golgi and Cajal, was working out the physical, cellular architecture of the tissue that electrical signal actually travels through, discrete, separate cells rather than one continuous net. It would take several more decades, well into the middle of the 20th century, before these two threads, the electrical mechanism and the cellular structure, were fully and rigorously combined into the single coherent picture of the neuron we walked through earlier tonight. But both threads needed to be pulled first, separately, by patient, careful researchers working with the limited tools available to them in their own particular moment, before anyone could even attempt to weave them together. The man in that kitchen was Camillo Golgi, an Italian physician working in the small town of Abbiategrasso, near Milan, in 1873. Golgi did not have a well-funded university laboratory at this point in his career. He was working in a converted hospital kitchen using basic equipment, largely on his own time, driven by pure scientific curiosity rather than any institutional mandate. He had been experimenting for some time with different chemical treatments for nervous tissue, trying to find something, anything, that would make individual structures visible under a microscope, and in 1873, he found a combination that worked in a way nothing had before. He took a piece of nervous tissue and hardened it using a solution containing potassium dichromate, and then treated it with silver nitrate. The chemistry of exactly why this worked wasn't fully understood even by Golgi himself, and in truth it still isn't perfectly understood even today, but the practical effect was extraordinary. The silver would, for reasons that seem to involve some kind of random precipitation process, deposit itself densely inside a small, seemingly random number of individual cells within the tissue sample, perhaps one cell in every 100, while leaving the vast majority of surrounding cells completely untouched, completely invisible, completely clear. The effect, when Golgi looked through his microscope, must have been startling. Where before there had only been an undifferentiated gray haze, now, scattered across the clear background, stood a small number of individual cells, each one stained a deep black, standing out with total clarity, every branch and extension of that single cell traceable by eye against the empty space around it, because none of the neighboring cells had picked up any stain at all. Golgi called this technique the black reaction, or reazione nera in his own Italian, and it remains, updated with modern variations, one of the most important staining techniques in the history of neuroscience, still used by researchers today, well over a century later. What Golgi had solved, essentially by accident, through years of patient trial and error, was the exact contrast problem that had been blocking progress on the deepest question in neuroscience. For the first time in human history, it was possible to look at a single nerve cell in isolation, to see its full shape, its branches spreading outward like a tree, or in some cases like the roots of a plant, without the branches of every other cell nearby cluttering the picture into unreadable noise. Here is the genuinely strange twist in this story, the part that makes it one of the most remarkable episodes in the history of science. Camillo Golgi, the very man who invented the technique that would finally answer the question of whether the brain was one continuous net or a population of separate cells, looked at his own beautiful clear images and concluded the wrong thing. Golgi remained convinced for the rest of his scientific career that the nervous system was one continuous, fused network, the reticular theory, exactly the view that most researchers of his era already held. He believed that despite what his stain revealed, the fine branches of different cells, though not directly visible as connected in his images, must actually fuse together at some point too small to see, even with his revolutionary technique, forming one single sprawling continuous structure after all. The evidence he had produced, arguably the clearest evidence anyone had ever produced on this exact question, was interpreted by its own inventor in a way that we now know was mistaken. This is worth sitting with for a moment, because it's a genuinely humbling fact about how science works. Having the right tool does not automatically mean you draw the right conclusion from what that tool shows you. Interpretation still requires a kind of intellectual leap, a willingness to trust what you see over what you already believe. And Golgi, brilliant as he was, could not quite make that leap himself. It would take a second man, working in Spain, using Golgi's own technique, to look at essentially the same kind of images and draw the opposite and correct conclusion. This was Santiago Ramón y Cajal, a Spanish physician and researcher working primarily in the 1880s and 1890s, and he is, by very wide consensus among historians of science, the single most important figure in the entire history of neuroscience. Cajal encountered Golgi's staining technique somewhat by chance, through a colleague who showed him some stained tissue samples in 1887, and Cajal was immediately captivated. He would later describe the impact of seeing this stained tissue for the first time as something close to a religious experience, a sudden opening of a door onto a world that had been completely hidden before. What set Cajal apart was not just access to the stain, since plenty of researchers across Europe were using Golgi's technique by this point. What set Cajal apart was a combination of extraordinary patience, an artist's eye trained through years of drawing as a younger man, and crucially, a refinement of Golgi's original method. Cajal developed his own modification of the black reaction, which produced even sharper, cleaner images, with individual cells standing out even more clearly against their background. He then spent years, an almost unimaginable volume of hours, at his microscope, examining stained tissue from many different regions of the brain, from many different species, and from animals at many different stages of development, including very young embryonic animals, where the branches of individual cells were simpler and less tangled, making them easier to trace clearly from end to end. And by working with young, developing tissue, where cells hadn't yet grown their full, dense adult complexity, Cajal was able to see something Golgi himself had missed. He could see, with real clarity, that the branches of individual nerve cells did not fuse together into one continuous structure. Instead, they came close to one another, at specific points of contact, but did not merge. Each cell remained its own separate, distinct, bounded unit, from beginning to end, touching its neighbors at particular points, but never physically becoming one continuous piece of material with them. This conclusion that the nervous system was made of individual, separate cells, communicating with each other at specific points of near contact rather than fusing into one continuous net, is what came to be known as the neuron doctrine. Cajal advocated for it forcefully through an enormous body of published work, and through strikingly beautiful, detailed hand drawings of what he observed under his microscope, drawings so precise and so scientifically valuable that many of them are still reproduced in neuroscience textbooks today, well over 100 years later, not as historical curiosities, but as genuinely useful, accurate depictions of real neural structure. Cajal was, by his own account and by all historical description, a relentlessly hard worker, often sketching late into the night, driven by what he described as a kind of obsession with understanding the deep hidden order of the nervous system, a conviction that if he simply looked carefully enough and drew what he saw with total honesty and precision, the true structure would reveal itself. The dispute between Golgi's continuous net theory and Cajal's neuron doctrine played out over more than 2 decades, through publications, through international scientific meetings, through direct and sometimes sharp disagreement between the two men, who, remarkably, had essentially never worked together and had developed their views largely independently, using variations of the very same staining technique. The scientific community gradually came to accept Cajal's interpretation as evidence accumulated, particularly as newer methods and better microscopes, developed in the decades that followed, continued to confirm what Cajal had argued, that nerve cells were indeed discrete, bounded, individual units. This was, remarkably, only fully and conclusively settled with certainty in the 1950s, more than half a century after Cajal's original work, when the invention of the electron microscope, an instrument capable of magnifying tissue far beyond what any light microscope could achieve, finally allowed scientists to see, with total unambiguous clarity, the actual physical gap, a real, measurable empty space, between the branch of one nerve cell and the branch of its neighbor. There was no fusion, there was a gap. Cajal had been right, and the electron microscope proved it beyond any possible doubt, decades after his death. In an unusual and rather poignant historical footnote, Golgi and Cajal were jointly awarded the Nobel Prize in Physiology or Medicine in 1906, honored together for their contributions to understanding the structure of the nervous system, despite holding fundamentally opposed views about what that structure actually was. In his Nobel lecture, delivered as part of the same ceremony where Cajal received his own prize, Golgi used the opportunity to argue once again, forcefully, against the neuron doctrine, restating his belief in a continuous net, even as the scientific tide was already turning decisively against him. It's a striking image, really. Two men sharing the same stage, honored for the same underlying body of technical work, one of them defending a picture of the brain that history would prove wrong, the other quietly certain he had found the truth, a certainty that later technology would fully vindicate. Now, here's the concept worth holding on to carefully, because it's the true foundation for absolutely everything else we'll cover tonight. Your brain is not one continuous, undifferentiated blob of thinking material, the way you might casually imagine gray matter to be if you'd never thought carefully about it. Your brain is a population, a genuinely enormous population, of individual cells called neurons, each one a separate, bounded, distinct unit, each with its own outer membrane holding it together as one discrete thing, each one capable of receiving signals, processing them in its own small way, and passing signals onward to other neurons. Picture it this way if it helps. Imagine not one continuous river of thought flowing smoothly through your head, but instead something more like an unimaginably vast city, built entirely of individual buildings, each building a single neuron, each one physically distinct and separate from its neighbors, connected to other buildings not by seamless walls but by countless individual wires and cables strung between them, each wire carrying its own message, at its own moment, to its own specific destination. And this discrete, cellular structure turns out to be the single most important fact underlying how learning and memory actually work, because it means that the connections between individual cells, the specific pattern of which building is wired to which other building, and how strong or weak each individual connection happens to be, can change. They can strengthen, they can weaken. New connections can form where none existed before. Old connections that go unused can fade away entirely. If the brain really were one single continuous fused structure, as Golgi believed until the end of his life, none of this kind of change, this fine, cell-by-cell, connection-by-connection modification, would even be a coherent possibility. There would be nothing to selectively adjust, no individual unit, and no individual connection to strengthen or weaken. It's precisely because the brain is made of discrete, separate cells meeting at specific points that experience can reshape it, gradually, connection by connection, across the whole span of a life, from the moment you're born until this very moment, right now, as new patterns of connection are quietly forming in your own brain in response to the very words you're hearing. Once the neuron doctrine was established, once it became broadly accepted that the nervous system was made of separate, individual cells rather than one continuous net, A new and pressing question opened up immediately. If neurons are separate, distinct units, not physically fused together, then how does a signal actually get from one neuron to the next? There has to be some kind of gap between them, since Cajal had shown that the branches of neighboring cells came close together without merging. So what happens in that gap? How does information actually cross it? The man who gave this gap its name was Charles Sherrington, an English physiologist working around the turn of the 20th century, roughly in the 1890s and early 1900s. Sherrington spent much of his career studying reflexes, the fast, largely automatic responses of the body to stimuli, such as the way your leg kicks forward when a doctor taps just below your kneecap with a small rubber hammer. Through very careful, methodical experiments measuring the timing and behavior of these reflex responses in animals, Sherrington noticed something important. The signal, as it passed through a chain of neurons involved in producing a reflex, did not travel with perfectly uniform, unchanging speed and strength the whole way through, the way it would if the whole pathway were one smooth, continuous, physically connected wire. Instead, there was a small but very real and consistent delay each time the signal had to cross from one neuron to the next, and the strength and behavior of the signal could actually be altered at each of these crossing points, sometimes boosted, sometimes reduced, sometimes blocked outright depending on other conditions. This was strong indirect evidence, before anyone could see the actual physical gap directly under a microscope, that something specific and significant was happening at the junction between one neuron and the next, some kind of active process, not just passive signal transmission down a continuous, unbroken wire. In 1897, Sherrington proposed the word synapse, drawn from a Greek term meaning to clasp or join together, to describe this junction point between neurons. He didn't yet know exactly what was happening physically at the synapse, but he had correctly identified, through careful indirect reasoning about timing and signal behavior, that something important, something worth naming and studying in its own right, was occurring at these countless individual junction points scattered throughout the nervous system. This left the field with a genuinely open and quite contentious question for the next several decades. What, physically, was actually crossing that gap at the synapse? Two broad camps emerged among researchers, and the disagreement between them came to be known, somewhat affectionately, in the history of neuroscience as the War of the Soups and the Sparks. One camp, the Sparks camp, argued that transmission across the synapse must be purely electrical, that whatever electrical signal traveled down one neuron simply jumped directly across the small gap to the next neuron, the way an electrical spark can sometimes arc directly across a small physical gap between two nearby wires. This view had a certain appeal, since it was already firmly established, going back to Galvani's work over a century earlier, that the basic signal traveling along a single neuron was electrical in nature. If the signal within a single neuron was electrical, it seemed reasonable, to many researchers, to assume that transmission between one neuron and the next would be electrical too, simply continuing on in the same basic form. The other camp, the Soups camp, argued instead that transmission across the synapse was chemical. In this view, when an electrical signal reached the very end of a neuron, right at the edge of the synaptic gap, it triggered the release of some kind of chemical substance, which then drifted physically across the tiny gap, and, on reaching the neighboring neuron, triggered a new electrical signal to begin there, effectively restarting the electrical process fresh in the next cell, rather than simply carrying the same electrical spark straight across empty space. This argument was settled, in a rather wonderful way, by an experiment that its own discoverer later said came to him in a dream, quite literally, while he was asleep. Otto Loewy, an Austrian physiologist, had been thinking hard about this exact synaptic transmission problem for years, without landing on a way to test it definitively, and then, according to his own later account, in 1921, he woke in the middle of the night with the complete design for a decisive experiment fully formed in his mind. He apparently scribbled some rough notes down on a scrap of paper by his bedside and went back to sleep, only to find, the following morning, that he could not read his own hurried, sleep-fogged handwriting well enough to reconstruct what he had been thinking. The story, as Lowy told it in later years, is that the very same dream returned to him again the following night, and this time, taking no chances, he got up immediately and went straight to his laboratory in the middle of the night to carry the experiment out right then, while it was still perfectly fresh and clear in his mind. The experiment itself was elegant in its simplicity. Lowy took 2 frog hearts and kept both of them alive and beating in separate containers of saline fluid, each heart connected to its own vagus nerve, a nerve that, among other functions, is known to slow the heart rate when it's stimulated. He electrically stimulated the vagus nerve attached to the first heart, and, exactly as expected, that first heart's beating slowed down. But then he took some of the fluid that had been surrounding that first stimulated heart, the fluid the heart and its stimulated nerve had been sitting in, and transferred it over into the container holding the second heart, a heart whose own vagus nerve had not been touched or stimulated at all. And the second heart's beating slowed down too, simply from being placed in that transferred fluid, with no direct electrical or physical stimulation of its own nerve whatsoever. This was very difficult to explain under the purely electrical, direct spark model of synaptic transmission, because no direct electrical contact of any kind had occurred between the stimulated heart and the second, resting heart. The only thing that had passed between them was fluid. Something chemical, something Lowy initially and simply called Vagusstoff, meaning vagus substance in German, had clearly been released into the fluid by the first heart's stimulated nerve, and that chemical substance alone, carried physically across into new fluid and then to an entirely separate heart, was sufficient to slow the second heart down as well. This chemical substance was later identified with certainty as acetylcholine, and it became the first neurotransmitter, the first chemical messenger of the nervous system, ever conclusively identified. Lowy shared the Nobel Prize in 1936 for this discovery, along with Henry Dale, a British scientist whose independent and complementary work had further confirmed and extended the same basic chemical picture of synaptic transmission. It's worth noting, as the field developed further across the 20th century, that both camps in this old war of soups and sparks actually turned out to be partly right, though the chemical, Soup's view proved by far the more common and more important mechanism throughout the vast majority of the brain. It's now understood that there are indeed also purely electrical synapses in the nervous system, direct physical channels connecting the inside of one neuron straight to the inside of a neighboring neuron, allowing electrical current to pass directly across, with essentially no delay at all. But these direct electrical connections are comparatively rare within the brain overall, and they're generally used in situations where extremely fast, tightly coordinated, synchronized activity across a group of neurons matters more than fine, flexible, adjustable control. The overwhelming majority of the truly enormous number of synapses in your brain, current estimates suggest something on the order of 100 trillion of them scattered throughout your nervous system right now, are chemical synapses, working through the release and detection of specific chemical messengers, exactly as Lowy's simple, elegant, 2-frog heart experiment first demonstrated back in 1921. One further piece of this puzzle, filled in a few decades after Lowy's original frog heart experiment, deserves mention here, because it directly explains something we relied on earlier tonight, without yet justifying it: the fact that some synapses excite the receiving neuron, while others inhibit it. This distinction was worked out with real precision by John Eccles, the same researcher who would later share the 1963 Nobel Prize alongside Hodgkin and Huxley, working through the 1950s using a technique that was, for its time, remarkably delicate, inserting an extremely fine glass microelectrode directly into the interior of a single living neuron, small enough not to destroy the cell in the process, and recording its voltage changes in real time as neighboring neurons were stimulated. Using this technique on neurons within the spinal cord, Eccles was able to directly observe and carefully distinguish two genuinely different kinds of response a receiving neuron could show following synaptic input. Some inputs produced what he termed an excitatory postsynaptic potential, a small, brief nudge of the receiving neuron's voltage toward its firing threshold, exactly the kind of nudge you'd expect from a synapse using glutamate. But other inputs produced the clear opposite, an inhibitory postsynaptic potential, a small nudge of voltage away from the firing threshold, actively working against the neuron firing, exactly the kind of effect you'd expect from a synapse using GABA. This was the first time anyone had directly recorded, from inside a single living neuron, clear, unambiguous electrical evidence that some synapses actively push a receiving neuron toward firing, while other synapses actively push it away, confirming with real electrophysiological precision what had previously been inferred more indirectly from the overall behavior of neural circuits. Eccles's careful intracellular recordings also settled, quite conclusively, the last significant piece of the earlier soups versus sparks debate we discussed earlier, since his direct measurements matched, in exact quantitative detail, what a chemical mechanism of transmission would be expected to produce, timing delays included, essentially proving beyond reasonable doubt that Lowy and Dale's chemical picture, rather than the earlier from-scratch purely electrical model, was the correct one for the overwhelming majority of synapses throughout the mammalian nervous system. This chemical mechanism turns out to matter enormously, for a reason that goes well beyond simply explaining how one neuron talks to another. Because transmission across the synapse is chemical, rather than a simple uniform electrical spark, it becomes something that can be finely, precisely modulated. A chemical signal can be released in larger or smaller amounts. It can be detected by a varying number of receptors, docking sites, on the receiving neuron's surface, and the number and sensitivity of those receptors can itself change over time, growing more numerous or less numerous, more sensitive or less sensitive, depending on the history of use at that specific connection. A purely electrical spark, jumping directly across a gap, wouldn't easily allow for this kind of graded, adjustable, endlessly variable behavior. But a chemical process with its many separate, tunable components, the amount of chemical released, the number of receptors present to detect it, the speed at which the chemical is cleared away afterward, and the connection reset, offers an enormous number of separate points where the strength of a single connection can be adjusted, gradually, up or down, based on experience. And that adjustability, sitting at the very heart of how a chemical synapse works, sitting at each one of those roughly 100 trillion individual connection points scattered through your brain right now, turns out to be the actual physical basis of learning and memory themselves, an idea we'll come back to and build out in real detail a little later tonight, once we've laid a bit more groundwork first. If you're still with me, let your eyes close now if they aren't already. You don't need to follow every word from here. Just let it wash over you. The explanation continues either way, and there's nowhere you need to keep up with. We're going to slow down even further now, and go inside a single neuron to see exactly how it works. Let's start with the shape of a single neuron, because the shape tells you almost everything about the function. A typical neuron has 3 basic parts, and it helps to picture something almost like a tree lying on its side, or perhaps a very unusual kind of tree with roots on one end and a single long trunk reaching out toward a distant patch of ground on the other. On one end, you have the dendrites, a branching tree-like structure that spreads outward and receives incoming signals from other neurons, thousands of them in some cases, arriving from all different directions, each one a separate small connection point. These dendrites gather together into the main body of the cell, called the soma, or cell body, which contains the cell's nucleus and the general machinery every living cell needs to sustain itself, to build proteins, to manage its own metabolism, to stay alive. And then, extending out from the soma in the opposite direction from the dendrites, is a single long, thin fiber called the axon, sometimes remarkably long, in some cases in your body stretching over a meter, from your spinal cord all the way down to a muscle in your foot, a single cell, one continuous living thread, spanning a meaningful fraction of your entire body's length. At the far end of the axon, the fiber branches out again into what are called axon terminals, and it's at these terminal branches that the neuron makes its connections, its synapses, onto the dendrites, or sometimes directly onto the cell body of other neurons further along. So information, in the most basic pattern, flows in one general direction through a neuron, in through the branching dendrites, gathered and combined at the cell body, then sent racing down the long axon, and finally passed onward, chemically, at the axon terminals, to the dendrites of the next neuron in the chain. Now let's slow down further and look at exactly how a signal moves along that axon, because this is one of the most beautiful and precise mechanisms in all of biology, and once you understand it clearly, you'll understand something genuinely fundamental about what is happening inside your own head, right now, as you listen. A neuron at rest, not currently firing a signal, maintains a small electrical voltage difference across its outer membrane, the thin wall that separates the inside of the cell from the fluid surrounding it. The inside of the cell, at rest, sits at a slightly negative voltage compared to the outside, typically around -70 millivolts, a genuinely tiny voltage by the standards of anything you'd notice in daily life, but very significant at the scale of a single cell. This resting voltage exists because of an uneven distribution of charged particles called ions, mainly sodium and potassium, across the cell's outer membrane. There's more sodium sitting outside the cell than inside, and more potassium sitting inside the cell than outside, and this uneven distribution is actively maintained, moment to moment, by tiny protein machines embedded in the cell membrane called ion pumps, which continuously work, spending energy the cell generates from food, to push sodium back out and pull potassium back in, constantly fighting against the natural tendency of ions to spread out evenly wherever they're free to move. Think of it a little like a dam holding back a reservoir of water at a higher level on one side than the other. The water isn't moving right now, but there's real, stored potential energy sitting there, built into the uneven arrangement, ready to be released the instant a gate is opened. And that's essentially what happens when a neuron fires what's called an action potential, the brief, sharp electrical signal that races down the axon and forms the basic unit of communication in the nervous system. When a neuron receives enough incoming signal from its dendrites, enough of a nudge, special gates in the cell membrane, called ion channels, specifically voltage-gated sodium channels, suddenly swing open at one point along the axon, right near the cell body. And when they open, sodium ions, which have been held back outside the cell by the resting voltage and the ion pumps working against them, come rushing rapidly into the cell, flowing down their own natural gradient, from an area of higher concentration outside to an area of lower concentration inside, the way water would rush eagerly through a suddenly opened dam gate. This inrush of positively charged sodium ions rapidly flips the local voltage inside the cell, at that one specific point along the membrane, from negative to positive, a very fast, very dramatic local voltage spike. Here is the elegant part, the part that makes this whole system work as a genuine, reliable, long-distance signal, rather than just a brief local flicker. This sudden local change in voltage, this spike, doesn't just happen and then sit there, fading quietly in place. It triggers the exact same kind of voltage-gated sodium channels sitting immediately next to it, just slightly further down the axon, to open as well, which causes the identical rapid sodium inrush and identical local voltage spike to occur at that next point too. And that, in turn, triggers the same thing to happen at the next point beyond that, and the next, in a self-propagating chain reaction that travels, quite rapidly, all the way down the full length of the axon, from where it starts near the cell body, all the way out to the distant axon terminals. This is why the whole event is called a wave, an action potential wave, propagating steadily and rapidly along the length of the fiber, regenerating itself fresh at every single point along the way, rather than gradually fading out the way an ordinary electrical signal, traveling passively through a simple wire, would naturally tend to weaken and dissipate with distance. Because the signal is actively regenerated at every point along its path, it arrives at the far end of even a very long axon at essentially full strength, exactly the same size and shape it had when it started, no matter how far it's had to travel. Right behind this traveling wave of activity, the cell doesn't just leave things flipped to their new, activated state. A slightly slower set of channels, potassium channels, opens shortly after the sodium channels, allowing potassium ions to flow back out of the cell, which helps restore the original negative resting voltage at that point along the membrane, resetting it, readying it to fire again later. And the sodium channels themselves, remarkably, also briefly enter a special inactivated state immediately after opening, a kind of very short built-in cooldown period during which they simply cannot be triggered to open again, no matter how strong the incoming signal might be. This brief unresponsive period is called the refractory period, and it turns out to matter enormously for how signals travel reliably through the nervous system, because it guarantees that the wave of activity can only ever travel forward, away from where it started, never backward toward a section of membrane that has only just fired and is still resetting. Without this simple built-in cooldown mechanism, you could get messy, chaotic signals sloshing back and forth uselessly along the axon, instead of one clean, reliable, one-directional pulse traveling cleanly from the cell body all the way out to the axon terminals, exactly where it needs to go. The two scientists who worked out this entire elegant mechanism in truly exacting, rigorous detail were Alan Hodgkin and Andrew Huxley, British physiologists working in the late 1940s and early 1950s. They conducted their key experiments using something called the squid giant axon, a genuinely enormous single nerve fiber found in certain squid species, remarkably large, in some cases close to a millimeter thick, which is astonishingly large for a single cell, and large enough that Hodgkin and Huxley could actually insert a fine measuring electrode directly down inside the axon itself and record the changing voltage happening right there, inside a single living cell, in real time as it fired, something that would have been essentially impossible using the vastly smaller neurons found in a mammalian brain, at least with the technology available at that particular moment in the mid-20th century. Through a long, painstaking series of careful experiments, systematically manipulating the concentration of different ions in the fluids surrounding the axon, and precisely measuring the resulting changes in voltage, Hodgkin and Huxley worked out the complete underlying mechanism we just walked through, describing it, remarkably, in a set of mathematical equations that could actually predict, with real precision, the exact shape, size, and speed of the action potential wave. Their work, published in 1952, remains one of the most complete and successful mathematical descriptions of a biological process ever produced, an achievement recognized with the Nobel Prize in 1963, shared with John Eccles, whose own work had helped clarify how signals were processed at the synapse itself, once the traveling wave finally reached its destination. It's worth appreciating the sheer speed and reliability of this whole system because it's easy to read a careful, slow, step-by-step description like the one we've just gone through and imagine something correspondingly slow happening inside your head. It isn't slow at all. A single action potential, from the moment it begins to the moment it completes, at one point along the membrane, takes roughly 1 to 2 thousandths of a second. And the wave itself, traveling the full length of an axon, can move at speeds ranging from around 1 meter per second in some neurons, up to over 100 meters per second in others, depending heavily on the specific structure of that particular axon, a detail we'll get to in just a moment. At the faster end of that range, a signal can travel from your spinal cord to your foot and, if needed, trigger a reflex response, in a small fraction of a second, fast enough that you can pull your hand away from a hot stove before the conscious, aware part of your mind has even finished registering, fully and clearly, that anything painful happened at all. The reflex arc for that kind of withdrawal typically doesn't even require your brain's higher processing centers to get directly involved in the initial decision to move. The spinal cord alone can often handle it, faster than full conscious awareness can even form. Now, that detail about axon structure affecting speed is genuinely worth pausing on, because it introduces another piece of the puzzle, another layer of engineering discovered inside the nervous system that dramatically improves how fast and how efficiently signals can travel. Many axons in your body, though notably not all of them, are wrapped, along much of their length, in a fatty, insulating substance called myelin, produced by specialized support cells that wind themselves tightly, layer over layer, around the outside of the axon, somewhat like tape wound carefully and repeatedly around a length of wire. This myelin coating is not laid down as one single continuous unbroken sleeve along the entire axon. Instead, it's applied in a series of distinct segments, with small, tiny, deliberate gaps left uncovered in between each segment. Gaps known as the nodes of Ranvier, named for the French anatomist Louis-Antoine Ranvier, who first carefully described this segmented structure back in the 1870s. Because the axon is insulated everywhere except at these small, regularly spaced gaps, the electrical signal can no longer regenerate itself smoothly and continuously at every single point along the membrane, the way it does in an uninsulated axon. Instead, the electrical change effectively leaps directly from one exposed gap to the next, skipping straight over the insulated sections in between, a process given the wonderfully descriptive name saltatory conduction, drawn from the Latin word for leaping or jumping. This leaping mechanism dramatically speeds up how fast a signal can travel down a myelinated axon, often by a factor of 10 or more, compared to an otherwise identical uninsulated axon of the same width, while also using considerably less of the cell's own metabolic energy to accomplish it, since the energy-intensive work of actively regenerating the full signal only needs to happen at each small gap, rather than continuously along the axon's entire length. This detail about myelin turns out to matter for a great deal more than pure engineering elegance. It's directly relevant to certain diseases, including multiple sclerosis, a condition in which the body's own immune system mistakenly attacks and damages the myelin coating surrounding axons in the brain and spinal cord, disrupting this efficient leaping form of signal transmission and causing the wide range of neurological symptoms associated with that particular disease. And it's also directly relevant to something happening constantly throughout ordinary human development, since the process of myelination, the gradual laying down of these fatty insulating wrappings around axons throughout the brain, continues actively for years after birth, in some brain regions continuing all the way into a person's mid-20s, gradually speeding up and refining neural communication as a person grows from infancy through childhood and well into full adulthood. One further wrinkle in this basic synaptic picture is worth adding here, because it shows how even something as ordinary and universal as pain isn't simply a fixed, direct readout of tissue damage arriving unfiltered into conscious awareness, but is instead actively shaped and adjusted at the level of the synapse by other information arriving at the very same time. In 1965, the psychologist Ronald Melzack and the physiologist Patrick Wall proposed what became known as gate control theory, suggesting that incoming pain signals traveling up the spinal cord toward the brain pass through a kind of gating mechanism at the level of the spinal cord itself, where they can be either amplified or dampened by other, simultaneously arriving signals, particularly ordinary touch signals carried along a different, faster class of nerve fiber. This theory offered a genuine physical explanation for something people had noticed anecdotally for a very long time without any clear mechanism to account for it. That briskly rubbing or pressing an area immediately around an injury, stubbing a toe, and instinctively grabbing and squeezing it, for instance, often provides real, immediate, if temporary, relief from the sharp pain of the injury itself. Under gate control theory, this happens because the fast-traveling touch signals generated by rubbing reach specific inhibitory neurons within the spinal cord and activate them, and those inhibitory neurons then release GABA, that same widespread inhibitory neurotransmitter we discussed earlier, directly onto the incoming pain pathway, at the very synapse where it would otherwise pass its signal onward and upward toward the brain, partially closing the gate and reducing how much of the original pain signal actually gets through. Gate control theory, refined and updated considerably since its original 1965 proposal, as more detailed knowledge of the specific spinal circuitry involved has accumulated, remains foundational to how modern pain management is understood, and it directly underlies certain non-drug approaches to pain relief still used in clinical practice today. including transcutaneous electrical nerve stimulation, which deliberately activates exactly this same touch-based gating pathway using mild electrical stimulation applied to the skin rather than through simple rubbing, in order to help dampen chronic pain signals through precisely the same basic synaptic mechanism. So, to bring this whole section together into one coherent picture before we move further out, a single neuron gathers incoming signals through its branching dendrites, combines and weighs all of that incoming information together at its cell body, and, if the combined incoming signal is strong enough to cross a certain threshold, fires off a rapid, self-regenerating, one-directional electrical wave, the action potential, that races down its axon, sometimes made dramatically faster and more efficient by a segmented fatty insulating coating, until it finally reaches the axon terminals at the very far end. And there, at those terminals, right at the very edge of the synapse, the entire nature of the signal changes completely, shifting from electrical to chemical, in a process we're going to look at closely right now, because this chemical handoff, happening at each one of the roughly 100 trillion synapses scattered throughout your brain, is genuinely where the real richness, flexibility, and endless adjustability of thought comes from. When the traveling electrical wave, the action potential, finally arrives at an axon terminal, it triggers the opening of yet another set of voltage-gated channels, this time ones that are specifically permeable to calcium ions rather than sodium. Calcium comes rushing into the very tip of the axon terminal, and this local rise in calcium concentration triggers small sacs called vesicles, tiny membrane-bound bubbles sitting just inside the terminal, each one already preloaded and waiting, full of a chemical neurotransmitter, to move toward the outer edge of the cell and fuse directly with the cell's outer membrane, spilling their neurotransmitter contents out of the neuron entirely, and into the narrow synaptic gap separating this neuron from the next one in the chain. This gap is astonishingly small, typically only around 20 nanometers across, meaning you could fit roughly 5,000 of these gaps, laid end to end, across the width of a single human hair, and yet, despite being separated by that tiny gap, the two neurons remain genuinely, physically distinct and separate cells the entire time, exactly as Cajal had first correctly argued, and exactly as the electron microscope would later confirm beyond any doubt. The released neurotransmitter molecules drift rapidly across this tiny gap through simple diffusion, and on reaching the other side, they bind to specialized receptor proteins embedded in the membrane of the receiving neuron, receptors shaped, quite precisely, to fit that particular neurotransmitter molecule, somewhat the way a specific key is shaped to fit a specific lock. When enough neurotransmitter molecules bind to enough of these receptors, they trigger a change in the receiving neuron, most commonly by causing tiny channels in its own membrane to open briefly, allowing charged ions to flow in or out at that specific location, and nudging that receiving neuron's own internal voltage either a little closer toward or a little further away from the threshold it would need to reach in order to fire its own action potential in turn. And here's a critical detail that adds enormous richness and flexibility to the whole system. Not every synapse pushes the receiving neuron in the same direction. Some synapses are excitatory, meaning the neurotransmitter released there nudges the receiving neuron's voltage a little closer to its firing threshold, making it somewhat more likely to fire soon. Others are inhibitory, meaning the neurotransmitter released there does the opposite, pushing the receiving neuron's voltage further away from its firing threshold, making it somewhat less likely to fire. The single most common excitatory neurotransmitter in your brain is a molecule called glutamate, while the single most common inhibitory neurotransmitter is one called GABA, short for gamma-aminobutyric acid. A single neuron in your brain, quite typically, receives thousands of these individual synaptic connections onto its dendrites at once, some excitatory, some inhibitory, arriving continuously from many different other neurons scattered across various different regions, and it is constantly, ceaselessly summing up all of this input together, moment to moment, weighing the total balance of excitation against inhibition, and it will only fire its own action potential when that carefully summed, constantly shifting balance, finally tips over its own specific firing threshold. This means a single neuron is not really behaving like a simple, dumb, passive relay switch, blindly passing every incoming signal straight along to the next cell. It's behaving much more like a small, continuous decision-making unit, constantly integrating a large number of competing excitatory and inhibitory inputs, arriving from many different directions and sources, and choosing, moment to moment, whether the balance of everything it's currently receiving is sufficient to justify firing its own signal onward to the next stage of the network. Multiply that single decision-making process by roughly 86 billion individual neurons, each one making its own small local decision, continuously, many times per second, all of them wired together through that staggering web of roughly 100 trillion individual synaptic connections. And you begin to get some real sense of the almost unimaginable complexity of what is happening inside your head, quietly, right now, as you drift, simply to let you hear, understand, and hold on to these very words. Before turning to critical periods specifically, it's worth stepping back briefly to consider how a brain gets built in the first place, since the process turns out to be considerably more dramatic and considerably more wasteful in a strangely productive way than most people imagine. In the earliest weeks of prenatal development, a specialized band of tissue called the neural tube begins folding itself into shape, and from that point onward, new neurons are produced at an extraordinary rate, with some estimates suggesting a developing human brain generates new neurons at a peak rate of several 100,000 per minute during certain stages of prenatal development. As newly formed cells migrate outward from where they're born to their eventual final position within the growing brain, guided along the way by specialized supporting cells and a rich array of chemical signals that help steer each migrating neuron toward roughly the correct destination. What happens after birth is, in some ways, even more surprising. Rather than simply continuing to add connections steadily throughout childhood, the young brain initially produces a considerable surplus, forming synaptic connections at a rate that, in the first few years of life, actually outpaces what will eventually be kept, reaching a peak density of synapses in certain regions of cortex sometime in early childhood that measurably exceeds the density found in a typical healthy adult brain. This is followed, across later childhood and continuing quite significantly through adolescence, by a long, gradual process of synaptic pruning, in which the connections that are used repeatedly and reinforced through actual experience are preserved and further strengthened, while the large surplus of connections that go relatively unused are steadily eliminated, a process in which those microglia we mentioned earlier as active immune cells play a genuinely important direct role. Physically, this overall pattern, overproduce first, then prune, based on experience, rather than growing only exactly what's eventually needed, turns out to be a remarkably efficient general strategy for building a brain that ends up well matched to the specific environment and set of experiences a particular individual actually encounters. Since it would be enormously difficult for a genetic blueprint alone to specify, in advance, the exact precise wiring diagram appropriate for every possible environment a person might eventually be born into. Producing a rich surplus and then refining it through lived experience solves that problem far more flexibly than trying to pre-specify the final answer from the start. This pruning process notably continues later, and for considerably longer, in the frontal lobe than in most other regions of the brain, with reliable evidence indicating that full structural maturation of frontal lobe circuitry, the very circuitry so dramatically and permanently disrupted in Phineas Gage's case, continues well into a person's mid-20s. This extended developmental timeline is now understood to be directly relevant to the well-documented pattern of somewhat greater impulsivity and heightened sensitivity to social and emotional reward, commonly observed during adolescence, a pattern that reflects a genuinely real, measurable difference in ongoing brain maturation. The frontal lobe's more deliberate, regulatory circuitry still actively developing and gradually catching up to earlier maturing regions involved in emotional and reward processing, rather than reflecting simply a matter of insufficient willpower or character, as it was long and rather unfairly assumed to be before this developmental research clarified the underlying picture. This raises a natural question. Is the brain equally plastic, equally open to change, at every point across a person's life? Or are there specific windows of time during which particular kinds of learning happen especially easily, perhaps even necessarily? The clearest, most influential answer to this question came from work conducted in the late 1950s and through the 1960s by David Hubel and Torsten Wiesel, working together at Harvard, studying the visual system of young kittens. Hubel and Wiesel discovered that if one eye of a very young kitten was temporarily sewn shut for a period during a specific early stage of development, depriving that eye of visual input during this particular window, the kitten would suffer a lasting, essentially permanent loss of functional vision in that eye once it was reopened, even though the eye itself, its retina, its lens, its optic nerve, remained completely physically healthy and undamaged throughout. The deficit was not in the eye, it was in the visual cortex, where they found, on carefully examining the neural tissue afterward, that columns of neurons, which should normally have been dedicated to processing input from the deprived eye, had instead been effectively taken over by neurons serving the other, unrestricted eye, as though the cortical territory belonging to the closed eye had simply been reassigned elsewhere during the period when it went unused. Crucially, this dramatic, largely irreversible effect only occurred when the eye was closed during a specific early window of development, later termed a critical period. Performing the exact same eye closure on an already mature adult cat produced no comparable lasting deficit at all, since by adulthood the relevant cortical wiring had already been established and locked in through normal early visual experience, and briefly withholding input no longer had the power to meaningfully reorganize it. This work, which earned Hubel and Wiesel a share of the Nobel Prize in 1981, alongside Roger Sperry's split-brain research, established the concept of the critical period as a genuine, biologically real phenomenon, not merely a loose figure of speech, and it turns out to apply well beyond vision alone. Human language acquisition shows a broadly similar, if less absolute and less sharply bounded pattern, with young children generally able to acquire the native-level pronunciation and grammatical intuition of a language with a natural ease that becomes measurably more difficult to fully replicate once a person is well into adulthood, even with intensive, deliberate effort, though adults certainly remain capable of learning new languages to a high level of practical fluency. What specifically becomes harder relates more to certain very fine-grained aspects of native-level processing than to language learning in any broad sense. This general pattern, early developmental windows, during which certain kinds of neural wiring are unusually sensitive to being shaped by experience, followed by a gradual narrowing of that particular sensitivity as the underlying circuitry matures and stabilizes, appears to be a genuinely important organizing principle across a range of different brain systems and functions, adding a further layer of nuance to the broader concept of neuroplasticity we discussed a little earlier. The brain remains capable of change throughout an entire life, but not every kind of change is equally easy to produce at every single stage of that life, and knowing roughly when a given system is at its most malleable turns out to matter quite a bit for understanding both normal development and, at times, for designing more effective interventions when development happens to go differently than expected. It's worth pausing on one more subtlety here, because it answers a natural question you might already be turning over. If a single action potential is always essentially the same fixed size and shape, the same all-or-nothing spike each and every time it fires, then how can a neuron communicate anything about intensity, about how strong or weak a particular sensation or signal actually is? A single fixed-size spike on its own can't easily carry that kind of graded information the way a smoothly varying signal could. The answer neurons rely on instead is timing and frequency rather than size. A neuron generally doesn't fire just once in isolation. It fires repeatedly, in a rapid train of individual spikes, and the rate at which it fires, how many spikes it produces per second, tends to scale up with the strength of the input driving it. A gentle touch against your skin right now might cause a relevant sensory neuron to fire at a modest, fairly low rate, while firmer pressure applied to that same patch of skin would cause that same neuron to fire considerably faster, a denser train of otherwise identical individual spikes, packed more tightly together in time. This general principle, called rate coding, is one of the most basic and well-established ways the nervous system represents the intensity of information, using a signal that is, at the level of any single individual spike, always the same fixed size. And on top of rate coding, researchers have also found evidence for a further layer of information carried in the precise relative timing of spikes, fired together by different neurons, sometimes called population coding or temporal coding, where the meaning of a signal depends not just on any single neuron's overall firing rate in isolation, but on the specific coordinated pattern across an entire group of neurons firing together, an area of active, ongoing research in its own right, and one that connects directly back to the binding problem we'll touch on again a little later tonight. Since coordinated, precisely timed firing across otherwise separate neurons and separate brain regions is one of the leading candidate mechanisms proposed for how the brain manages to bind together separately processed pieces of information into one single, seamless, unified experience. There's one more piece worth adding to this picture before we move on, because it explains how a synaptic signal, once it's done its job, doesn't simply linger there forever, endlessly and uselessly re-triggering the receiving neuron over and over. After a neurotransmitter has bound to its receptors and done its work, it needs to be cleared away fairly quickly so the synapse can reset and be ready to respond freshly to the next incoming signal. This clearing happens through a few different mechanisms depending on the specific neurotransmitter involved. Some neurotransmitters are broken down directly in the synaptic gap by specific enzymes. Acetylcholine, the very same neurotransmitter Otto Lowy identified in his dream-inspired frog heart experiment, is cleared this way, broken apart by an enzyme called acetylcholinesterase. Other neurotransmitters, including glutamate and GABA, are instead pulled back up and reabsorbed directly into the neuron that originally released them, or sometimes absorbed by nearby support cells through specialized reuptake proteins sitting in the cell membrane, ready to be repackaged into fresh vesicles and used again later. This reuptake process, incidentally, happens to be a major target of several well-known classes of medication, including many antidepressants, which work in large part by slowing down the reuptake of certain neurotransmitters, allowing them to linger somewhat longer in the synaptic gap than they normally would, and thereby increasing their overall effect on the receiving neuron over time. Now that you have a clear picture of how a single neuron works, how it fires, how it hands its signal chemically across a synapse to the next cell in line, it's time to zoom back out and look at how these basic units get organized into the larger structures that actually let you do the enormous range of things a human brain does: seeing, remembering, feeling, moving, planning, dreaming. This is where the story branches out considerably, because the same basic building block, the neuron, firing and connecting in fundamentally the same way everywhere in your body, gets organized into wildly different regional architectures, each specialized for a different kind of job. Let's begin with the broadest possible layout, the one you'd see if you simply looked at a whole human brain from the outside. The largest, most visible part, the wrinkled, folded outer layer that most people picture when they imagine a brain, is called the cerebral cortex, and its heavily folded, crumpled appearance is not accidental or arbitrary. Those folds, the ridges called gyri and the grooves between them called sulci, exist because folding allows a much larger total surface area of cortical tissue to be packed into the limited volume of your skull, similar to how crumpling a large flat sheet of paper into a ball lets you fit far more paper into a small box than you could if you insisted on keeping it perfectly flat. If you could carefully unfold and flatten out an average adult human cortex, smoothing away all its natural creases, it would cover an area somewhere in the range of a large placemat, roughly 2,500 square centimeters, and yet this entire folded structure is normally only somewhere between 2 and 4 millimeters thick, a thin, richly wrinkled sheet of neurons wrapped snugly around the rest of the brain. The cortex itself is conventionally divided into 4 major regions on each side, called lobes, and each one, broadly speaking, specializes in a somewhat different category of function, though it's important to say clearly that almost nothing in the brain is handled by just one isolated region, working entirely alone. Nearly every real mental function you can name draws on rich, extensive cooperation between many different areas, working together at once. Still, the broad map is genuinely useful. At the very back of your head sits the occipital lobe, dedicated very heavily to processing visual information arriving from your eyes. Along the sides, roughly around and above your ears, sit the temporal lobes, heavily involved in processing sound and language, and also housing structures deeply important for forming new memories, which we'll get to shortly. Toward the top and back sits the parietal lobe, which integrates sensory information, particularly touch and the sense of where your own body is positioned in space, and helps guide your spatial awareness and attention. And at the very front, behind your forehead, sits the frontal lobe, which is heavily involved in planning, decision-making, impulse control, and what's often loosely called executive function, the general capacity to hold a goal in mind and organize your ongoing behavior around achieving it. Much of what's known about this particular region's role in personality and impulse control traces back to one of the most famous and unusual cases in the entire history of medicine, that of Phineas Gage, a railroad construction foreman working in Vermont in 1848. Gage was using a long iron tamping rod to pack explosive powder into a hole drilled in rock, in preparation for blasting, when the powder detonated prematurely, driving the rod, over a meter long and more than 3 centimeters thick, straight up through his left cheek, behind his eye, and out through the top of his skull, landing a considerable distance away, heavily coated in blood and brain tissue. Remarkably, Gage did not die, and did not even lose consciousness for more than a very brief period. He was able to speak within minutes, and was, by most accounts, sitting upright and talking within the hour, a fact that astonished the physicians who treated him. He went on to survive for roughly another 12 years. What made the case so significant for the future study of the brain was not the survival itself, remarkable as it was, but the specific, lasting change in Gage's personality that his physician, John Martin Harlow, carefully documented afterward. Before the accident, by all available accounts, Gage had been a responsible, well-liked, even-tempered foreman. Afterward, according to Harlow's own written description, Gage became markedly more impulsive, irritable, and unable to stick with previously made plans, prone to profanity he had not shown before, and generally so changed in temperament that friends and acquaintances reportedly said he was no longer Gage. The rod had passed almost entirely through his frontal lobe on the left side, leaving most of his other cognitive abilities, including memory, language, and basic movement, largely intact, while apparently damaging, fairly specifically, the circuitry involved in planning, impulse control, and social judgment. Later reconstructions of the case, using modern imaging techniques applied to Gage's preserved skull, which is still held today at Harvard's Warren Anatomical Museum, have refined the original account considerably, and some of the more dramatic popular retellings of Gage's personality likely overstate what the historical record can actually support with full confidence. But even with that appropriate caution, the case remains one of the earliest and most influential pieces of evidence connecting specific frontal lobe damage to specific, lasting changes in personality and impulse control. A connection that later, far more carefully controlled research, including detailed studies of other patients with more precisely documented frontal lobe injuries, has gone on to confirm and refine considerably. The very back portion of the frontal lobe, right along the boundary with the parietal lobe, contains a narrow strip called the motor cortex, which sends out the commands that ultimately produce voluntary movement throughout your body, while a parallel narrow strip just behind it, right at the front edge of the parietal lobe, called the somatosensory cortex, receives incoming touch information from across your entire body surface. One of the more delightful and genuinely useful discoveries about these two strips is that each one is organized as a rough, distorted map of the entire body, laid out continuously along the strip from one region to another. But the map is far from evenly proportioned. Body regions with especially dense, fine sensory or motor control, particularly your hands, your lips, and your tongue, take up a hugely disproportionate amount of space on these cortical maps compared to body regions like your back or your calves, which, despite covering considerably more actual physical surface area on your real body, receive comparatively little dedicated cortical territory. This mapping was worked out in painstaking detail in the 1930s and 1940s by a Canadian neurosurgeon named Wilder Penfield, who, while performing brain surgery on fully conscious patients, a standard and genuinely necessary practice at the time for certain kinds of epilepsy surgery since the brain tissue itself has no pain receptors and patients can safely remain awake and responsive throughout, would gently stimulate different points along these strips with a small electrode and simply ask the patient, in real time, what they felt or noticed happening in their own body. Patient by patient, region by region, Penfield built up an extraordinarily detailed and remarkably consistent map, sometimes illustrated as a strange, distorted little figure known as the cortical homunculus, with enormous hands and lips and a comparatively tiny torso, a vivid visual reminder that the space your brain dedicates to any given body part reflects how much fine-grained sensory or motor detail that part requires, not how physically large that part happens to be. Beneath this large, folded outer cortex sit a number of smaller, deeply important structures, tucked away further inside the brain, structures that don't get nearly as much everyday public attention as the cortex, but that are absolutely essential to how the whole system functions. One of the most important of these is the hippocampus, a small, curved structure, roughly the size and shape of a seahorse, which is in fact exactly where its name comes from, drawn from the Greek word for seahorse. The hippocampus plays an absolutely central role in forming new long-term memories, particularly what are called episodic memories, memories of specific personal events and experiences, things that happened to you, at a particular place, at a particular time. We know just how central the hippocampus is to this specific function, largely because of one of the most studied and genuinely tragic cases in the entire history of neuroscience. A patient known for decades in the scientific literature only by his initials, HM, whose full name, Henry Molaison, was only made public after his death in 2008. In 1953, as a young man suffering from severe disabling epilepsy that had resisted every other available treatment, Molaison underwent an experimental surgery in which surgeons removed large portions of his hippocampus on both sides of his brain, in an effort to control the source of his seizures. The surgery was, in a narrow sense, successful in reducing his epilepsy, but it produced a devastating and completely unanticipated side effect. Molaison lost the ability to form almost any new long-term memories at all, a condition called anterograde amnesia. He could still recall much of his life from before the surgery reasonably well, and he could hold a normal conversation in the present moment just fine, using what's called working memory, the brain's capacity to hold a small amount of information active and available for a matter of seconds at a time. But the moment his attention shifted away from something, it was essentially gone from him forever. He would meet the very same researcher, someone who had worked closely and directly with him for years, day after day, and be introduced to them, fresh, as a complete stranger, every single time, with absolutely no memory of ever having met them before, no matter how many hundreds of times the meeting had already happened. Molaison worked closely with researchers, most notably Brenda Milner, for decades following his surgery, and the extensive, careful study of his condition became foundational to essentially everything neuroscience now understands about how the hippocampus specifically supports the formation of new long-term memories, and, just as importantly, about the fact that different kinds of memory clearly depend on genuinely different brain structures. since Molaison's ability to learn certain new physical skills, such as a mirror tracing task in which you have to trace a shape while only seeing your hand's movement reflected backward in a mirror, actually kept improving, session after session, even though he had absolutely no conscious recollection whatsoever of ever having practiced the task before. This dissociation, seen clearly and directly in a single, extensively studied patient, is a large part of why neuroscience now recognizes multiple genuinely distinct systems of memory, rather than treating memory as one single, unified faculty. Sitting close by the hippocampus is another small but critically important structure called the amygdala, an almond-shaped cluster of neurons deeply involved in processing emotion, and particularly fear. The amygdala receives incoming sensory information rapidly, and it can trigger an emotional response, especially a fear response, remarkably quickly, in some cases even before the higher, more deliberate reasoning centers in your cortex have fully finished consciously processing exactly what's happening. This is part of why you might flinch violently away from a coiled shape on a hiking trail before you've consciously registered whether it's actually a snake or simply a curled piece of rope, a genuinely useful evolutionary shortcut for situations where a slightly premature but fast reaction is, on balance, considerably safer than a slower but more perfectly accurate one. The amygdala also plays an important role in attaching emotional weight and significance to memories as they're being formed, which is a large part of why emotionally intense experiences, both wonderful and terrible ones, tend to be remembered far more vividly and far more durably than ordinary, emotionally neutral events. Deeper still, closer to the very center of the brain, sit a pair of structures called the thalamus, which acts as a kind of major relay and routing station, receiving incoming sensory information from nearly every one of your senses, with the notable exception of smell, and passing that information onward to the appropriate specific regions of the cortex for further, more detailed processing. And below the thalamus sits the hypothalamus, a comparatively tiny structure, yet one that plays an outsized role in regulating an enormous range of basic bodily functions: body temperature, hunger, thirst, and, importantly for tonight, the sleep and wake cycle, working in close, coordinated partnership with that small pineal gland Descartes once mistakenly believed was the seat of the soul, which, as we covered earlier, actually produces melatonin in response to the hypothalamus's ongoing tracking of the daily pattern of light and darkness. The hypothalamus also sits at the very top of a longer, slower signaling chain worth understanding, since it directly governs how your body handles stress, and understanding it helps explain why an anxious, racing mind can feel so physically difficult to simply switch off at bedtime. When the brain registers something as a genuine threat or a significant stressor, the hypothalamus releases a chemical signal that travels a very short distance to a small gland sitting just beneath it, the pituitary gland, which in turn releases its own signal into the bloodstream, this one traveling considerably farther, all the way down to the adrenal glands, sitting atop each kidney, prompting them to release the hormone cortisol into general circulation throughout the body. This whole pathway, known as the hypothalamic pituitary adrenal axis, or HPA axis, operates on a fundamentally different timescale than the action potentials and synaptic signals we spent most of tonight discussing. Where a single action potential completes in roughly 1 to 2 thousandths of a second, and a neurotransmitter is cleared from a synaptic gap within a similarly small fraction of a second, this hormonal stress pathway unfolds over minutes, sometimes considerably longer, since it depends on a chemical messenger physically traveling through the bloodstream to reach a distant target organ, rather than crossing a synaptic gap measured in mere nanometers. Cortisol, once released, has wide-ranging effects throughout the body, including increasing available blood sugar for quick energy and heightening overall alertness, effects that were, over the long course of evolution, genuinely useful for responding to an immediate physical threat, but that can become considerably less useful when the same pathway is triggered repeatedly by the kind of chronic, ongoing psychological stress common in modern daily life, unrelated to any actual physical danger, requiring a burst of immediate energy. And this same stress pathway connects directly back to the sleep-related material we covered earlier tonight, since elevated cortisol works in direct, active opposition to the calming, sleep-promoting signals sent by melatonin, which is a large part of the physiological explanation for why a mind still actively cycling through the day's stresses, worries, or unresolved problems can feel so much harder to settle at night than a mind that's genuinely at ease, and why deliberately slowing down exactly the way you're doing right now, simply by lying still and letting your attention rest on a calm, unhurried voice, can measurably help shift this whole balance back in favor of rest. Toward the back and base of the brain sits the cerebellum, a densely folded structure that, despite taking up only around 10% of the brain's total volume, is estimated to contain more than half of all the neurons in the entire brain, packed into tightly, densely folded tissue. The cerebellum is heavily involved in coordinating smooth, precisely timed movement, and in a related, more subtle role in certain kinds of procedural learning and fine-tuned timing more broadly, including some aspects of learning new physical skills, not unlike the improving mirror tracing skill that Henry Molaison, despite his profound memory impairment, was still able to acquire. Before we leave the topic of chemical messengers, it's worth going a little deeper into the variety of neurotransmitters at work in your brain, because glutamate, GABA, and acetylcholine, the three we've discussed so far, are only part of a considerably larger cast of characters, and several of the others are directly responsible for some of the most familiar and consequential aspects of everyday human experience, including mood, motivation, and attention. Consider dopamine, a neurotransmitter produced by a relatively small cluster of neurons deep in the midbrain, in a region called the ventral tegmental area, whose axons nonetheless project widely outward to many other parts of the brain, including regions involved in movement and, especially, a set of structures often referred to collectively as the reward system. For a long time, dopamine was popularly described, somewhat imprecisely, as the brain's pleasure chemical, the thing released whenever you experience something enjoyable. More careful research conducted from the 1990s onward, much of it led by the neuroscientist Wolfram Schultz working with monkeys trained on simple learning tasks, revealed something more interesting and considerably more useful than a simple pleasure signal. Schultz found that dopamine neurons fire most strongly, not simply when a reward arrives, but specifically when a reward arrives unexpectedly, or more precisely, when it arrives in a way that violates the animal's prior expectation about how likely or how large that reward was going to be. If a monkey learned to reliably expect a reward following a particular cue, the dopamine response gradually shifted, over repeated trials, away from the moment the reward itself was delivered, and toward the earlier moment when the predictive cue first appeared, as though the brain's excitement had migrated backward in time to attach itself to the earliest available signal that reward was coming. And if an expected reward was then withheld entirely, dopamine neurons showed a distinct dip, firing below their normal baseline rate, precisely at the moment the reward should have arrived but didn't. This pattern, now generally described as a reward prediction error signal, suggests dopamine functions less like a simple pleasure meter and more like a constantly updating teaching signal, informing the rest of the brain, moment to moment, whether outcomes are turning out better or worse than expected, and thereby helping to guide future learning about which actions and cues are actually worth pursuing. Consider also serotonin, produced primarily by a cluster of neurons in the brainstem called the raphe nuclei, again with projections reaching widely throughout the brain, influencing mood, appetite, and sleep regulation among other functions. Serotonin's role in mood regulation became the basis for one of the most widely prescribed categories of medication in modern medicine, the selective serotonin reuptake inhibitors, or SSRIs, which work by partially blocking the reuptake process we described earlier tonight, the process by which a neurotransmitter, once released, gets pulled back up into the neuron that originally released it. By slowing this reuptake specifically for serotonin, these medications allow serotonin to remain active in the synaptic gap somewhat longer than it otherwise would, which appears, over a period of weeks rather than immediately, to produce beneficial effects on mood for many people experiencing depression, though it's worth being honest that the precise, complete mechanism by which this eventually translates into improved mood remains, even now, an area of active ongoing research, since the immediate biochemical effect on serotonin levels happens far faster than the clinical improvement in mood typically does, a mismatch in timing that researchers are still working to fully explain. And consider norepinephrine, produced by a small brainstem structure called the locus ceruleus, Latin for the blue spot, so named for its slightly bluish appearance caused by the pigment inside its neurons, which plays a central role in regulating arousal, alertness, and the classic fight-or-flight stress response, working in the brain in a manner closely related to how the hormone adrenaline, sometimes called epinephrine, operates throughout the rest of the body. And finally, briefly, consider the endorphins, a category of naturally occurring chemical messengers, structurally related to opioid drugs like morphine, that your own brain produces internally in response to pain and vigorous physical exertion, contributing to the pain-dampening, sometimes mood-lifting effect that people commonly experience after sustained physical exercise. Each of these systems, dopamine, serotonin, norepinephrine, and the endorphins, uses the exact same basic underlying cellular mechanism we spent so much of tonight walking through carefully: action potentials, vesicles, and receptor binding across a synaptic gap, simply tuned to a different specific messenger molecule and wired up to a different specific pattern of connections throughout the brain, which is a large part of why so much of the enormous range of human mood, motivation, and experience can ultimately be traced back to the same handful of basic biological building blocks, arranged and connected in different ways. Now let's turn to something you've likely already heard mentioned casually before, sometimes with more confidence than the underlying science actually supports: the idea of the left brain and the right brain. It is genuinely true that the brain is divided into two roughly mirror-image hemispheres, left and right, connected to one another by a thick band of connecting fibers called the corpus callosum, which allows constant, rapid, extensive communication to flow back and forth between the two sides. And it is also genuinely true that certain specific functions do show a reasonably reliable degree of lateralization, meaning they tend to be handled somewhat more heavily by one hemisphere than the other. In the large majority of people, most language processing, including both the physical production of speech and much of its grammatical comprehension, is handled predominantly by the left hemisphere, a pattern first carefully documented in the 1860s by the French physician Paul Broca, who studied patients with damage to a specific region of the left frontal lobe, now known as Broca's area, who lost most of their fluent capacity to physically produce speech, while nonetheless retaining a reasonably good ability to understand language spoken to them by others. A related but importantly distinct language deficit was documented shortly afterward, in the 1870s, by the German physician Karl Wernicke, whose patients showed damage to a different specific region, this one in the left temporal lobe, now known as Wernicke's area, and who could speak quite fluently, often at considerable length, but whose speech tended to be largely nonsensical, and who also had serious, genuine difficulty understanding language spoken to them. Some of the clearest and strangest evidence for lateralization came from a series of studies conducted in the 1960s on a small number of patients who had undergone a surgical procedure called a corpus callosotomy, in which the corpus callosum, that thick connecting band of fibers linking the two hemispheres, is deliberately cut, again as a last resort treatment for otherwise unmanageable severe epilepsy, in order to stop electrical seizure activity from spreading freely from one hemisphere into the other. These so-called split-brain patients, studied extensively by the psychobiologist Roger Sperry and his then graduate student Michael Gazzaniga, could function remarkably normally in ordinary daily life following the surgery, but under carefully controlled experimental conditions, researchers were able to briefly present visual information to only one half of a patient's visual field at a time, ensuring that the information reached only one hemisphere, since each hemisphere primarily processes the opposite side of the visual field, and, because the connecting fibers had been severed, that information could no longer be shared over to the other hemisphere the way it normally would be. The results were remarkable. If an image of an object was flashed briefly only to a split-brain patient's left visual field, meaning the information arrived only in the right hemisphere, the patient would typically report seeing nothing at all, since the left hemisphere, which handles most language production in the great majority of people, had received no information whatsoever about what had just been shown. And yet, if that same patient was then asked to reach with their left hand, which is controlled by the right hemisphere, behind a screen and select the matching object from a group of hidden items by touch alone, they could often do so correctly, despite being unable to verbally name or describe what they were selecting or why. It was, in effect, as though two separate streams of processing, only loosely aware of one another, were operating within a single body at once, each hemisphere possessing genuine knowledge the other hemisphere simply had no access to. Sperry received the Nobel Prize in 1981 for this body of work, and it remains some of the most direct and startling evidence available that the two hemispheres, while normally working in such constant, seamless cooperation that a person never notices any division at all, are nonetheless capable, under these very specific surgical circumstances, of processing and even acting on information genuinely independently of one another. But the popular idea, so common in casual conversation, that some people are simply left-brained, meaning rigidly logical and analytical, while other people are simply right-brained, meaning free-flowing and creative, as though this described two fundamentally different overall personality types, is a considerable oversimplification of the real underlying science, not well supported by careful modern brain imaging research. Nearly every complex mental task you can name, including tasks popularly associated primarily with just one hemisphere, such as language or spatial reasoning, actually engages substantial activity across both hemispheres working together in close, constant coordination, not one hemisphere operating in some kind of isolation from the other. The real, well-established lateralization findings, such as language leaning left in most people, are genuine and scientifically solid, but they describe a modest relative specialization within a system that remains deeply and continuously interconnected across both sides, not two separate, independently operating brains housed side by side inside one skull. Now, let's turn to a concept that ties directly back to everything we established earlier about the synapse, and that finally gives us the real physical mechanism underlying learning and memory, the thing we promised to return to. The key underlying idea here is called synaptic plasticity, meaning the capacity of an individual synapse, one single connection point between two specific neurons, to change in strength over time, becoming either more effective or less effective at influencing the receiving neuron, depending directly on the pattern and history of activity that has passed through it. The foundational theoretical idea behind this was proposed by Donald Hebb, a Canadian psychologist, in 1949, well before anyone had any direct experimental tool capable of actually measuring changes in synaptic strength directly. Hebb proposed, based mostly on careful theoretical reasoning about how learning ought to work, that when one neuron repeatedly and persistently takes part in firing another neuron, some kind of lasting change should occur at the specific connection between them, a change that makes that particular connection more efficient going forward, more likely to successfully trigger the second neuron the next time the first one fires. This idea is very often summarized today with the shorthand phrase, neurons that fire together wire together, and while that's a considerable simplification of Hebb's original, more careful reasoning, it captures the essential core of the idea reasonably well. Direct experimental evidence for exactly this kind of change was found decades later, in 1973, by Timothy Bliss and Terje Lermo, who were studying a particular neural pathway within the hippocampus of an anesthetized rabbit. They found that if they stimulated a particular set of incoming fibers with a brief but intense burst of high-frequency electrical activity, then the response of the receiving neurons to ordinary, everyday strength stimulation of that very same input pathway remained measurably and durably stronger afterward, for hours, and in some further experiments, for days or even considerably longer, compared to how that same pathway had responded before the intense burst. They named this lasting strengthening effect long-term potentiation, very often shortened simply to LTP, and it remains, to this day, the single most well-studied and most widely accepted physical candidate mechanism underlying how the brain actually forms and stores lasting memories at the level of individual synaptic connections. The physical molecular details of how long-term potentiation actually works involve, in significant part, that glutamate receptor system we touched on earlier. A particular kind of glutamate receptor, called the N-MINDA receptor, behaves in an unusually clever way. It typically stays effectively blocked, unable to let ions flow through it freely, unless two separate specific conditions are both satisfied at the exact same time. The receiving neuron generally has to already be somewhat electrically activated on its own, and, separately, glutamate specifically needs to be actively binding to that same receptor at that very same moment. This dual, both conditions required requirement, effectively makes the N-MINDA receptor behave almost like a tiny biological coincidence detector, only truly springing open when the sending neuron and the receiving neuron happen to be active together, at essentially the same moment in time, which lines up beautifully and precisely with Hebb's original theoretical proposal from decades earlier. When this N-MINDA receptor does open under these specific joint conditions, it allows a meaningful pulse of calcium to enter the receiving neuron, and that calcium pulse in turn triggers a whole cascade of further molecular changes inside the cell, including, notably, the insertion of additional glutamate receptors of a different specific type into the receiving neuron's membrane, right at that exact synapse, which makes that particular connection measurably more sensitive and more responsive to future incoming signals arriving along that same specific pathway, essentially strengthening that one connection while leaving neighboring, uninvolved connections on the very same neuron completely unaffected. There's an important, precisely opposite process too, called long-term depression, or LTD, in which certain specific patterns of activity, rather than strengthening a synapse, instead weaken it over time, reducing its future influence on the receiving neuron. This might sound, on first hearing, like a purely negative or undesirable process, but it's every bit as essential to healthy brain function as strengthening is. Without some active mechanism for weakening unused or unhelpful connections, a brain would gradually and inevitably become saturated, with every connection eventually strengthening toward its own maximum possible level, and no further room left for any new or more useful learning to be reflected in the pattern of connections. The healthy, effective operation of memory and learning genuinely depends on this constant, ongoing, dynamic balance between strengthening some connections and weakening others, continuously reshaping the fine-grained pattern of synaptic weights across the whole brain based on ongoing experience, moment to moment, day to day, across an entire lifetime. This capacity for connections to change is very often described using the broader term neuroplasticity, and it's worth being genuinely clear and precise about what this term does and does not mean, since it gets used rather loosely in a lot of popular, non-scientific writing. Neuroplasticity does not mean that the brain can effortlessly rewire itself into literally any configuration at any time, nor does it mean that any single skill can always be picked up with equal ease at any age or stage of life. One particularly well-documented and genuinely striking real-world illustration of this involves London taxi drivers, who, in order to earn their official license, are required to memorize an enormously detailed mental map of London's notoriously complex, sprawling street layout, a rigorous and demanding process that typically takes several years of dedicated, focused study to complete. Researchers using brain imaging found that experienced London taxi drivers who had successfully completed this demanding training showed measurably larger volume in the posterior portion of their hippocampus, the specific rear part of that structure particularly associated with detailed spatial memory and navigation, compared to the general population, and further, that this measured volume difference correlated directly and reliably with the number of years each individual driver had actually spent doing the job. This is strong, concrete evidence that sustained, demanding mental practice over an extended period can produce genuine, measurable structural change in the adult human brain, change visible directly on a brain scan, not merely some vague, purely metaphorical figure of speech about learning. One further discovery worth including here, both for how it happened and for what it revealed, involves a category of neuron identified almost entirely by accident in the early 1990s in the laboratory of the Italian neurophysiologist Giacomo Rizzolatti at the University of Parma. Rizzolatti's team was recording the activity of individual neurons in the motor cortex of macaque monkeys, studying neurons involved in specific hand movements, such as grasping a piece of food, a fairly standard kind of motor cortex research. The by now famous, if somewhat informally recounted story, is that a researcher in the lab happened to reach out and pick up a piece of food themselves, near a monkey that was sitting still, not moving at all, while still connected to the recording equipment, and the electrodes registered a burst of activity in a neuron that had previously only fired when the monkey itself performed that same grasping action. Follow-up, carefully controlled experiments confirmed this was a real and repeatable effect, and not simply a one-off fluke. Certain neurons in this region of the motor cortex fired both when the monkey performed a specific goal-directed action itself and when the monkey merely observed another individual, whether monkey or human, performing that same action. Rizzolatti's group named these mirror neurons, and the discovery generated an enormous amount of excitement and speculation, since it suggested a possible neural mechanism by which observing someone else's action could be at least partly represented inside an observer's own brain, using much of the same neural machinery the observer would use to perform that action personally, offering a tempting, physically grounded candidate explanation for a range of human abilities involving understanding other people, including imitation learning, empathy, and even, in some more speculative extensions of the theory, language itself. It's important to be honest, though, that the field's understanding of mirror neurons has grown considerably more cautious and measured since those early, highly publicized findings. And this makes for a genuinely useful case study in how enthusiasm for a striking initial result needs to be balanced against slower, more careful follow-up work. Direct single neuron recording of the kind Rizzolatti's team performed in monkeys is far more difficult to carry out in humans for ethical reasons, so most human evidence for a comparable mirror system relies on indirect methods, including fMRI studies showing overlapping regions of activation during both performing and observing an action, evidence that's suggestive but considerably less precise and direct than recording individual neurons. And a number of researchers have pushed back specifically against some of the broader, more ambitious claims occasionally made about mirror neurons single-handedly explaining complex human capacities like empathy or language, arguing that these more sweeping claims run well ahead of what the underlying evidence actually, carefully supports. The core finding itself, that some neurons in certain motor regions respond both to performing and to observing a matching action, remains genuinely well established. But precisely how large a role this specific mechanism plays in the broader, richer human capacity for understanding and connecting with other people's actions and intentions remains, quite genuinely, an open and actively debated question, and it offers a good reminder, worth carrying with you through the rest of tonight, that even a discovery as striking and evocative as this one deserves the same careful, patient, skeptical follow-up that every claim in this field has ultimately needed, going all the way back to Aristotle's cooling radiator and Golgi's own stubborn belief in a continuous net. Alongside neurons themselves, it's worth taking a moment to properly credit an entirely separate category of brain cell that doesn't fire action potentials or form the kind of synapses we've spent most of tonight discussing, but that is nonetheless completely essential to how the whole system functions. These are the glial cells, sometimes still casually and rather unfairly described, based on outdated older thinking, as merely the brain's structural glue, which is in fact exactly where their name comes from, drawn from the Greek word for glue. Glial cells actually outnumber neurons in the brain by a significant margin, and they perform an enormous range of genuinely essential jobs. Some, called oligodendrocytes, are specifically responsible for producing that myelin insulation we discussed earlier, wrapping tightly and repeatedly around axons to dramatically speed up signal transmission. Others, called astrocytes, help regulate and clean the chemical environment immediately surrounding synapses and are now understood to play a considerably more active and direct role in modulating synaptic activity and strength than earlier generations of researchers had ever suspected, essentially serving as active partners in neural communication rather than simply passive structural support scaffolding. Still others, called microglia, act as the brain's dedicated resident immune cells and are now understood, through more recent research, to play a genuinely important active role in pruning away weak or unused synaptic connections, quite literally helping to physically clear away and eliminate the connections that long-term depression has already weakened. A kind of active, ongoing maintenance and refinement process that helps keep the overall network efficient, well-organized, and free of unnecessary cluttering unused connections. Much of what we've described in this last stretch, the mapping of function to specific brain regions, the study of plasticity in living adult brains, the observation of dopamine neurons responding to reward, depends on tools that let researchers observe brain activity without cutting the skull open at all. And it's worth taking a moment to appreciate where those tools came from, since they represent their own separate, fairly recent revolution in how the brain can be studied. The earliest of these tools traces back to a German psychiatrist named Hans Berger, who, in 1924, first recorded measurable electrical activity from the surface of an intact human scalp, using electrodes placed against the skin and a sensitive recording device to detect the faint combined electrical signal produced by the coordinated activity of enormous populations of neurons firing together underneath. Berger called this recording an electroencephalogram, or EEG, and though his early results were met with real skepticism, partly because Berger himself was somewhat reclusive and slow to publish extensive supporting detail, other researchers eventually replicated and confirmed his findings, establishing EEG as a genuinely useful window into the brain's ongoing electrical activity. EEG cannot tell you precisely which specific neurons are firing, or exactly where in three-dimensional space inside the brain a given signal originates, since it's measuring a blurred, combined signal passing through the skull and scalp from countless neurons at once. But it offers something no other common technique can match nearly as well: extremely precise timing, capable of detecting changes in brain activity down to a matter of milliseconds, fast enough to track the brain's electrical rhythms as they shift, moment to moment, including the specific rhythmic patterns that define different stages of sleep, which we'll return to shortly. A considerably newer and quite different tool is functional magnetic resonance imaging, or fMRI, developed and refined mainly through the 1990s, building on the magnetic resonance imaging technology already established in medicine more broadly. Rather than measuring electrical activity directly, fMRI takes advantage of a more indirect signal. Active neurons require more oxygen, delivered through the blood, and so regions of the brain that are working harder at any given moment draw in a slightly greater supply of oxygenated blood shortly afterward. Because oxygenated and deoxygenated blood have very slightly different magnetic properties, a sufficiently powerful and sensitive magnetic resonance scanner can detect these small regional differences in blood oxygen level, producing a three-dimensional map of which brain regions were relatively more or less active during a given task. This gives fMRI a real strength that EEG lacks, considerably more precise spatial detail about where in the brain activity is occurring, but a real corresponding weakness too, since the blood flow response it's actually measuring lags behind the underlying neural activity by a few seconds, making fMRI far better suited to answering questions about which brain regions are involved in a given kind of task than to tracking the millisecond by millisecond timing of neural processing the way EEG can. The two techniques in this sense are genuinely complementary, one offering fine-grained timing with blurry spatial detail, the other offering fine-grained spatial detail with blurry timing, and much of modern neuroscience research draws on both, along with several other, more specialized techniques, including positron emission tomography, or PET, which uses a small amount of injected radioactive tracer to measure metabolic activity or the distribution of specific neurotransmitter receptors directly, and magnetoencephalography, or MEG, which detects the extremely faint magnetic fields produced by electrical activity in the brain, offering a combination of good timing precision and somewhat improved spatial detail compared to standard EEG, though at the cost of requiring considerably more expensive and specialized equipment, generally housed in a heavily magnetically shielded room. And finally, worth mentioning here is the considerably more recent and, for a long time, genuinely controversial discovery of adult neurogenesis, meaning the ongoing birth of entirely new neurons from scratch in specific, limited regions of the fully adult human brain. For most of the 20th century, the firm, confident textbook consensus among neuroscientists was that humans are born with essentially every single neuron they will ever have across their entire lifetime, and that no meaningful new neuron growth occurs anywhere in the brain after fairly early childhood development. This turns out to be at least partially incorrect. Research beginning in the 1990s and continuing steadily since then has found reasonably solid evidence for at least some new neuron formation continuing throughout adult life, specifically within that same hippocampus we discussed earlier, in a small specific substructure called the dentate gyrus. Though the exact overall extent and the true ongoing significance of this adult neurogenesis in humans specifically remains an area of genuinely active scientific research and some continued careful debate even today, a nice, honest example of a real scientific question that is still, quite openly, in the process of being actively worked out, rather than something settled long ago and simply waiting to be reported. Let's bring all of this back down to where you actually are right now, lying there in the dark, with your own brain doing all of this very moment, without any conscious effort from you at all. Start with your phone, probably resting somewhere nearby on a nightstand or under your pillow, since so many of us keep it close even at night. There's a genuinely direct connection between the device sitting there and the brain we've spent tonight describing, because the field of artificial intelligence that powers so much of what your phone does, the systems that recognize your voice, suggest the next word in a text message, or even generate a voice much like the one speaking to you right now, drew real, substantial, historical inspiration from this same basic picture of the neuron we just walked through. The artificial neural networks used throughout modern computing are built from units loosely modeled on the same basic principle Cajal first correctly identified. Many simple individual processing units, each one receiving multiple weighted inputs, summing them together, and producing an output only once some threshold is crossed, then passing that output along to other connected units in turn. The artificial version is a drastic, enormously simplified abstraction of the real biological mechanism, missing almost all of the rich chemical, temporal, and structural complexity we've covered tonight. But the basic underlying inspiration, connectionism, the idea that complex intelligent behavior can emerge from a large network of very simple interconnected units, adjusting their connection strengths through experience, traces back directly to the neuron doctrine that Cajal fought for over 100 years ago. Consider the simple fact of the blanket or sheet resting against your skin right now. Something so ordinary you likely aren't even consciously registering it as a sensation at all. And yet it turns out that exactly how your body converts that physical pressure into a nerve signal in the first place was only worked out with real molecular precision within the last couple of decades, a genuinely recent piece of this whole long story. For a very long time, researchers knew, in general terms, that specialized nerve endings in the skin respond to touch and pressure, but the exact molecular mechanism, the specific protein responsible for actually converting physical mechanical force into an electrical signal, remained unidentified. This was finally resolved in 2010, when the researcher Ardem Pataputian and his laboratory, working through a systematic process of testing and eliminating candidate genes one at a time in cultured cells that had been engineered to respond mechanically to touch, identified a protein they named piezo-1, and shortly afterward a closely related second protein called piezo-2. both of which form a specialized channel directly embedded in the cell membrane of certain touch-sensing neurons. A channel that physically opens, allowing ions to flow through and trigger a nerve signal, specifically in response to mechanical deformation of the membrane itself. Essentially a microscopic pressure-sensing gate built directly into the shape of the protein. Around the same general period, a separate but related line of research, led by the researcher David Julius, worked out the molecular mechanism behind how your body senses temperature, using a wonderfully direct and clever experimental approach, testing which specific receptor protein, among many candidates, was activated by capsaicin, the compound responsible for the burning sensation associated with chili peppers. Julius's laboratory identified a receptor, now called TRPV1, that opens in response to both capsaicin and to genuinely elevated temperature, which is exactly why eating a sufficiently spicy chili pepper produces a sensation your brain interprets as heat, even though no actual rise in tissue temperature has occurred, since the very same molecular gate that responds to real heat also happens to respond to this particular plant compound. Julius and his collaborators went on to identify a related receptor responsible for sensing cold in a broadly similar way. Julius and Pataputian jointly received the Nobel Prize in Physiology or Medicine in 2021 for this body of work, and it's a genuinely fitting one to mention here tonight specifically, since the very last stretch of this whole 2,000-year story, from the Edwin Smith papyrus, noting a pulsing wound with no explanation at all the way to a precise, named molecular gate responsible for the exact feeling of a blanket resting against your own skin right now, was worked out well within the past 15 years. Genuine reminder that this field, for all its considerable age, is still actively, rapidly filling in real, previously unknown detail, even in areas as seemingly basic and universal as the simple sensation of touch. Think too about what's happening in your own body as you simply lie there, still, breathing slowly, listening. Your hypothalamus, that small but powerful structure we described earlier, has been tracking the falling light levels in your environment throughout the evening, and in response, it has been signaling your pineal gland, that very same structure Descartes once mistakenly believed to be the seat of the human soul, to steadily increase its production of melatonin, gradually easing your whole body towards sleep. Your body temperature is very likely dropping slightly right now, as part of this same natural process, a small physical shift that itself helps facilitate the transition into sleep. Your heart rate is probably slowing as well, gradually shifting under the growing influence of what's called your parasympathetic nervous system, the general resting and restorative counterpart to the more familiar fight-or-flight response. All of it ultimately coordinated and orchestrated by circuits within your brainstem and hypothalamus, working quietly and continuously in the background, entirely without any conscious involvement or decision-making from you at all. Consider, too, the room around you, the walls, the ceiling, whatever soft shapes and shadows are visible in the low light. Your visual system, even in near darkness, is still processing whatever faint patterns of light happen to be reaching your closed or open eyes, sending that information back through your thalamus and onward to your occipital lobe, where it's assembled, quite automatically, into a coherent, unified sense of the surrounding space you occupy, even though you may not be paying close conscious attention to any of it right now. That whole visual pathway, remember, is the very same basic system Alcmaeon first began to trace over 2,000 years ago in ancient Croton, tracking the optic nerve as it ran from the eye toward the brain, one of the very first specific instances anyone ever recorded of a physical nerve pathway carrying information from the outside world directly into the body's central processing organ. And your own ears, right now, picking up the sound of this voice, whatever device it's coming through, are converting the physical vibration of air molecules into an electrical signal through a genuinely remarkable piece of biological engineering. Tiny hair cells inside your inner ear bending in response to sound waves and triggering nearby auditory neurons to fire, sending signals up through your brainstem and thalamus into your temporal lobes, where language processing regions not too far from Wernicke's original 19th century discovery are actively working, right now, to parse the individual words in this sentence into meaning, all of it happening through exactly the same basic mechanism of action potentials and chemical synapses we spent so much time carefully walking through earlier tonight, applied here specifically to sound and language rather than to touch or movement or memory. It's worth mentioning, too, that the very same basic question Cajal spent his career trying to answer with a microscope and a steady hand, exactly which neuron connects to which other neuron, is still being pursued today, only now at a scale Cajal himself could scarcely have imagined, using tools built specifically for this purpose. This modern effort goes by the name connectomics, aimed at producing a complete, exhaustive wiring diagram, a connectome, mapping every single synaptic connection within a given nervous system. The first such complete map was published in 1986 for a small roundworm called Caenorhabditis elegans, whose nervous system contains a mere 302 neurons in total, a number small enough that a dedicated team of researchers, led by Sidney Brenner and John White, working over roughly a decade, could trace essentially all of its connections by hand, using thousands of electron microscope images of extremely thin tissue slices, painstakingly reconstructed and cross-referenced. Even at that comparatively tiny scale, the achievement was considered a landmark, since it represented the very first time any organism's complete wiring diagram had ever been fully mapped out from beginning to end. Scaling this same basic approach up to a mammalian brain, let alone a full human brain with its roughly 86 billion neurons and 100 trillion synapses, represents an almost unimaginably larger undertaking, but recent advances in automated high-speed electron microscopy and machine learning-assisted image analysis, itself in a nice bit of circularity, built on artificial neural networks originally inspired by the biological neuron, have made real, substantial progress possible. In 2023, an international collaborative research effort successfully published a complete connectome of the fruit fly brain, containing roughly 140,000 neurons and tens of millions of synaptic connections, an effort that would have been essentially unthinkable using Cajal and Golgi's original hand-tracing methods, but that automated modern imaging and computational reconstruction made achievable within a few years of concentrated, well-resourced effort. Mapping a complete human connectome remains, for now, a considerably more distant goal, given the sheer difference in scale, but smaller mapped portions of mammalian brain tissue, including detailed reconstructions of small cubic millimeter samples of mouse and even human cortex, have already been completed and published, and they continue to reveal genuinely new and sometimes unexpected structural details about how real neural circuits are actually organized, well beyond what could ever have been inferred purely from the kind of large-scale regional maps Penfield or Broca and Wernicke were able to produce. It's a fairly direct, unbroken line, in other words, from Golgi's accidental discovery of the black reaction in a converted hospital kitchen in 1873, to a modern research team's automated microscope quietly imaging an entire fly brain, slice by impossibly thin slice, 150 years later, still chasing, with vastly better tools now available, exactly the same basic question Cajal was chasing by candlelight and hand-drawn sketch. Which cell talks to which other cell, and how? There's something worth noticing in the sheer scale of coordination required for something as ordinary and unremarkable as simply lying still and listening to a voice. Nothing about this moment feels effortful to you. It doesn't feel like work. And yet, underneath that effortless surface sits an almost incomprehensibly vast amount of continuous, coordinated cellular activity, sensory information arriving continuously through your ears and eyes, hormonal and neural systems steadily easing your body towards sleep, memory systems in your hippocampus quietly encoding fragments of tonight's explanation, even as your conscious attention drifts in and out, emotional processing systems in your amygdala, perhaps registering the general sense of comfort or calm associated with this particular nightly routine, if this is something you do regularly. All of it running simultaneously, all of it built from the same basic unit, the neuron, and the same basic mechanism, the synapse, that took human beings literally thousands of years, from the embalmers of ancient Egypt discarding brains they thought were worthless, all the way to Cajal hunched over his microscope late into the night in Spain, to finally understand even in outline. Think also about memory itself and how it's likely to work on you specifically, tonight, and in the days following. Because your hippocampus is going to keep working on tonight's material even after you've fully fallen asleep, and even after this recording has ended. A substantial and growing body of sleep research indicates that memory consolidation, the process by which a fresh, fragile, recently formed memory gets gradually stabilized and reinforced into a more durable, lasting form, happens significantly during sleep itself, particularly during certain specific sleep stages. Some of the very same neural activity patterns that occurred while you were originally learning something new have actually been observed recurring, replaying in a compressed, accelerated form during subsequent sleep, essentially your brain quietly rehearsing and reinforcing today's experiences while you rest. This is a large part of why getting genuinely adequate sleep matters so much for learning and memory generally, and it's a rather fitting thought to end this particular section on, since it means that whatever you do or don't consciously remember from tonight's explanation by tomorrow morning, some quiet trace of it, some small adjustment to the pattern of synaptic strength somewhere in your own hippocampus, may well be getting gently reinforced and consolidated right now, precisely while you sleep. It seems fitting, given what this particular channel is for, to spend a little more time on what's actually about to happen to your own brain over the next several hours, because sleep itself, once thought to be simply a passive shutting down of brain activity, a kind of nightly power-off, turns out to be nothing of the sort. This was first demonstrated clearly using that same EEG technology Hans Berger developed back in 1924, when, in 1953, a graduate student named Eugene Aserinsky, working under the physiologist Nathaniel Kleitman at the University of Chicago, was monitoring the eye movements and brain activity of sleeping research subjects, including, in the earliest observations, his own young son. Aserinsky noticed periods during the night when the sleeper's eyes moved rapidly back and forth beneath their closed lids, and when he checked the corresponding EEG recordings from these same periods, he found something that must have seemed puzzling at first. The brain's electrical activity during these episodes looked, in its general pattern, remarkably similar to the brain's electrical activity during ordinary, alert wakefulness, fast and relatively low in amplitude, rather than the slower, larger, more synchronized waves typically associated with deep sleep. Waking subjects during these specific periods and asking what they had just experienced confirmed what this odd combination of signs suggested. This was the stage of sleep during which vivid dreaming most reliably occurs. Aserinsky and Kleitman named it rapid eye movement sleep, or REM sleep, and their 1953 publication marked the real beginning of sleep as a serious, rigorously measurable subject of scientific study, rather than a topic left mostly to guesswork and folk belief. Since that original discovery, researchers have mapped out a fuller picture of what a typical night's sleep actually involves, and it turns out to be considerably more structured and eventful than the simple, undifferentiated word sleep tends to suggest. A typical night moves through repeating cycles, each cycle lasting roughly 90 minutes, and within each cycle, the sleeper passes through several distinct stages. Early in each cycle come the lighter stages of what's called non-REM sleep, during which EEG recordings show a gradual slowing and increasing synchronization of brain activity as the sleeper moves from light drowsiness into progressively deeper sleep. The deepest of these non-REM stages, sometimes called slow-wave sleep, is marked by large, slow, highly synchronized electrical waves sweeping across broad areas of the cortex together, and this particular stage appears to be especially important for physical restoration and for certain kinds of memory consolidation, including the kind of factual, declarative memory consolidation relevant to material like what you've been hearing tonight. Then, later in each 90-minute cycle, the sleeper moves into REM sleep, that lighter, more wakefulness-resembling stage Aserinsky first identified, during which the body's major voluntary muscles are actively, deliberately paralyzed, a genuinely useful protective mechanism preventing a dreaming sleeper from physically acting out whatever their dreaming brain happens to be depicting, while the eyes themselves move rapidly and the brain generates the vivid, often narratively strange experiences we call dreams. As the night progresses through its several repeating 90-minute cycles, the balance gradually shifts, with deep, slow-wave sleep dominating more heavily earlier in the night, and REM sleep occupying a progressively larger share of each cycle as morning approaches, which is part of why dreams tend to feel more vivid and memorable, specifically in the hours just before natural waking. The purpose of REM sleep specifically, and of dreaming more generally, remains, even now, one of the genuinely open questions in this field, with several serious competing hypotheses rather than one single settled answer. Some researchers emphasize REM sleep's apparent role in processing and regulating emotional experience, based partly on studies showing that emotionally difficult memories seem to lose some of their sharp emotional intensity, specifically after a period including adequate REM sleep, as though the brain were using this particular stage to file away the factual content of an experience, while gradually softening its raw emotional charge. Others emphasize REM sleep's role in a particular kind of creative, associative memory consolidation, pointing to research suggesting that people are sometimes measurably better at solving problems requiring novel, unusual connections between pieces of information after a period of sleep that included substantial REM, compared to an equivalent period spent purely awake. Still others focus on more basic proposed functions, including the ongoing maintenance and calibration of the visual system, given the close involvement of rapid eye movement itself. It's entirely possible, and in fact seems reasonably likely, that REM sleep serves several of these purposes simultaneously, rather than any single one exclusively. But a fully complete, settled account remains, honestly still out of reach. One more example, alongside the ones we're about to turn to directly, of a genuinely open question sitting right at the heart of an otherwise well-studied and well-understood system. Now let's turn, gently, to some of the genuinely open, unresolved questions that scientists studying the brain still find themselves debating today, because it seems only honest, after walking through so much settled, well-established science, to spend a little time on the parts that remain, quite genuinely, unsettled. The single deepest and most stubbornly unresolved question in this entire field is usually referred to as the hard problem of consciousness, a phrase coined by the philosopher David Chalmers in the 1990s. The so-called easy problems of consciousness, though calling them easy is itself something of a joke, since they remain enormously difficult in their own right, involve figuring out which specific brain processes correlate with specific mental states, which regions become active when you see the color red, or feel pain, or recall a childhood memory. Difficult as they are, these questions are at least the right shape for scientific investigation, since they involve tracing measurable physical correlations between brain activity and reported mental experience. The hard problem is different and considerably stranger. It asks why any of this physical activity, all those ion channels opening, all that calcium rushing into axon terminals, all those vesicles releasing their chemical contents into all those countless tiny synaptic gaps, should be accompanied by any subjective experience whatsoever. Why isn't all of this simply happening in the dark, so to speak? Complex information processing occurring with no inner experience attached to it at all, the way it presumably occurs in a calculator or a very sophisticated computer. Why does it feel like something to be you, specifically right now, rather than all of this elaborate biological machinery simply running along, without any subjective feeling accompanying it whatsoever? Nobody currently has anything close to a fully satisfying answer to this question, and there is genuine, ongoing, active disagreement among serious researchers and philosophers about whether it's even a question science, in its current form, is actually capable of eventually answering at all, or whether it might require some genuinely new kind of conceptual framework we simply don't have yet. A closely related and equally difficult puzzle is often called the binding problem. Different aspects of a single unified experience, the color of an object, its shape, its motion, the sound it might be making, are all processed we know from careful research, in genuinely separate, physically distinct regions of your brain, often processed with slightly different timing and even at slightly different processing speeds relative to one another. And yet you don't experience the world as a scattered, disconnected collection of separate features arriving on separate channels. You experience it as one single, seamlessly unified scene. Something, somewhere, is binding all of these genuinely separate streams of neural processing back together into one coherent, unified experience. And precisely how that binding actually happens, mechanically, remains a genuinely open and actively debated question. Some researchers have proposed that synchronized, precisely timed firing across different, otherwise separate brain regions might be part of the answer. Groups of neurons in different areas firing together, in careful rhythm, as a way of effectively tagging their signals as belonging to the very same underlying object or event. But this remains, even now, an active area of vigorous ongoing research, rather than a fully settled, textbook conclusion. There's also considerable genuine debate about the true nature and full extent of neuroplasticity, the very topic we covered a little earlier tonight. While the basic underlying reality of adult brain plasticity is now firmly, solidly established, well beyond serious scientific doubt, there remains real, active disagreement about exactly how much functional reorganization is genuinely possible in specific circumstances, particularly following serious brain injury, and about precisely which underlying mechanisms account for the sometimes quite dramatic recoveries occasionally observed in certain patients. Some of the most striking documented cases involve young children who have undergone a hemispherectomy, the surgical removal or disconnection of an entire cerebral hemisphere, typically as a last resort treatment for severe, otherwise untreatable epilepsy, who nonetheless go on, in some cases, to develop remarkably functional language and motor abilities using only their single remaining hemisphere. Precisely how much of this striking recovery reflects the young brain's genuinely superior plasticity compared to an older adult brain, and how much instead reflects functions that may already have existed, in some more limited or latent form, on both hemispheres from a very early stage of development, even before the surgery took place, remains a real and active subject of ongoing scientific investigation and discussion. A related and equally unsettled debate concerns the nature of voluntary decision-making itself, sometimes framed, in its boldest form, as a question about free will. In the early 1980s, a physiologist named Benjamin Libet ran a genuinely striking experiment. He asked participants to perform a very simple voluntary action, flexing their wrist, whenever they personally felt the spontaneous urge to do so, with no external cue or instruction about timing at all, and he asked them to note, using a specially designed fast-moving clock, the precise moment they first became consciously aware of their own intention to move. Meanwhile, he recorded their brain activity using EEG electrodes, watching for a well-established electrical signature called the readiness potential, a slow buildup of activity over motor planning regions of the cortex that reliably appears in the brief period leading up to a voluntary movement. Libet found that this readiness potential reliably began building, on average several hundred milliseconds before participants reported becoming consciously aware of their own intention to move, meaning some measurable brain activity associated with preparing the movement appeared to be underway before the participant's own conscious experience of deciding to act. Libet's original interpretation, and the interpretation that made the experiment so famous and so widely discussed well outside neuroscience itself, was that this sequencing raised a serious challenge to the intuitive, everyday sense that conscious intention is what initiates a voluntary action, since here, at least in this specific simple task, the brain's preparatory activity seemed to come first, with conscious awareness of the intention arriving only afterward. This result, and the many variations and refinements of it conducted by other researchers in the decades since, remains genuinely contested. Some researchers have argued the early readiness potential reflects general background neural noise rather than a specific decision already made, with the actual decision only crystallizing later, closer to the reported moment of conscious awareness. Others have questioned how reliably participants can actually judge the precise timing of their own inner experience using a fast-moving clock in the first place, given how notoriously difficult introspective timing judgments are to make accurately under laboratory conditions. And still others accept Libet's basic finding, but argue it doesn't undermine free will, so much as it complicates the popular, oversimplified idea of a single conscious moment cleanly initiating every action, suggesting instead that conscious awareness may function more as a kind of gatekeeper, capable of vetoing or allowing an already forming impulse, rather than as the original spark that starts it. More than 4 decades after the original experiment, this remains a live, actively debated topic, drawing together neuroscientists, psychologists, and philosophers without anything close to full consensus. There's also a more sobering open question worth mentioning honestly, given how directly it affects real people, despite the very detailed picture we've built up tonight of how a healthy neuron and a healthy synapse work. A full, precise understanding of exactly what goes wrong in neurodegenerative conditions like Alzheimer's disease remains, even now, incomplete. It's been known for decades that Alzheimer's disease is closely associated with the abnormal accumulation of 2 particular proteins in the brain, one called amyloid beta, which tends to clump together outside neurons into dense deposits known as plaques, and another called tau, which under healthy conditions helps stabilize the internal structural scaffolding inside a neuron, but which, in Alzheimer's disease, becomes abnormally modified and tends to collapse into tangled clumps inside the cell itself, known as neurofibrillary tangles. For a long time, the dominant working theory in the field held that amyloid accumulation was the primary root cause driving the whole disease process, triggering the later tau tangles and the resulting widespread neuron loss and memory impairment. This amyloid-focused theory shaped an enormous amount of research funding and drug development for decades, yet a series of high-profile clinical trials of medications specifically designed to clear amyloid from the brain have produced results that were, at best, modest, and at times genuinely disappointing relative to the strength of the underlying theory, leading a considerable number of researchers in the field to argue for a more complex, multi-factor picture of the disease, one that may involve chronic inflammation, blood vessel health, the immune system's own resident brain cells, those microglia we mentioned earlier as active pruners of synapses, and quite possibly a more complicated, still not fully mapped relationship between amyloid, tau, and the actual loss of synaptic connections that most directly and immediately correlates with the memory decline patients and families experience. This remains, quite genuinely, one of the most actively contested and consequential open questions in all of neuroscience today, precisely because getting the answer right carries such enormous, direct human stakes for the millions of people currently living with the disease, and for the families supporting them. And finally, there's the question of memory's own underlying reliability, a question with real, direct, practical consequences well beyond pure scientific curiosity. Careful research conducted over the past several decades, much of it associated with the psychologist Elizabeth Loftus, has shown quite clearly and repeatedly that human memory does not function anything like a simple, fixed video recording, accurately and permanently capturing a past event exactly as it originally happened, and then simply playing it back later, unchanged, whenever it's recalled. Instead, every single act of recalling a memory appears to involve a genuine process of reconstruction, actively rebuilding the memory fresh, from underlying component pieces, each and every time it's retrieved. And this reconstruction process turns out to be genuinely and measurably vulnerable to distortion, to the quiet influence of new information encountered after the original event, and even, in some carefully controlled experimental circumstances, to the outright implantation of entirely false memories for events that demonstrably never actually happened at all. Exactly how and why memory works this particular reconstructive way, rather than functioning as a simple, stable, faithful playback system, the way many people naturally and intuitively assume it does, and precisely what this reveals about the deeper underlying purpose evolution originally shaped memory to serve, remains a genuinely rich and actively studied area of ongoing research, with real implications extending well beyond the laboratory, including into how legal systems around the world think about and evaluate the reliability of eyewitness testimony in criminal courts. None of these open questions should feel unsettling. If anything, they're a kind of quiet comfort in their own way. An organ this old, this thoroughly studied, across so many centuries, by so many careful and dedicated people, still holds real, genuine mystery at its very core. There's still discovery left to be made. There's still room for wonder, even after all of tonight's explanation. By now you may have drifted in and out a few times. That's exactly right. There's nothing to catch up on. Everything has been settling quietly, whether you were following closely or not, gathering gently into whatever shape it's going to take by morning. If you were to hold all of tonight in one image, it would be this. Picture the smallest possible unit first. One single neuron, quietly holding a small negative charge across its own thin outer membrane, patiently waiting, gathering thousands of tiny incoming whispers from its neighbors along its branching dendrites, some pushing it gently toward action, some holding it gently back. And then, when the balance finally tips, that neuron answers with the same clean, reliable, self-renewing signal it always uses, a wave of opening and closing gates racing down its own long fiber, arriving finally at its very tip, where it converts itself, seamlessly, from an electrical signal into a chemical one, releasing its small burst of message into an almost impossibly narrow gap, to be caught, on the other side, by another separate, distinct whole cell, never fused with the first, always separate, exactly as Cajal insisted it must be, against Golgi's own belief, against 1400 years of Galen's hollow tubes, against Aristotle's cooling radiator, against every reasonable, careful, mistaken guess that came before. Now multiply that single unit by roughly 86 billion. Let each one sit embedded in its own small local neighborhood, and yet also reach out, through the choreography of that same 100 trillion connections, into wider circuits, circuits shaped like tightly folded sheets of cortex mapping the surface of your own body in strange, distorted proportion, hands and lips enormous, back and calves nearly forgotten. Circuits curled like tiny seahorses, quietly filing away the specific, personal texture of what actually happened to you today, distinct from the separate circuits, tucked lower and further back, that instead hold the simple motor memory of how to ride a bicycle or trace a shape correctly in a mirror. Skills that persisted, remarkably, even in a man who could no longer recognize the very people teaching them to him, every single day, as if for the first time. Circuits shaped like an almond, ready to flinch you away from a curled rope on a trail before you've even consciously decided it might be a snake. Circuits deep at the center, quietly tracking the fading light outside your window right now, nudging a small gland Descartes once mistook for the seat of an immaterial soul to release the very hormone easing you, cell by cell, toward sleep. And running underneath every single one of those circuits, holding the entire structure together, supplying it, insulating it, pruning it when it goes quiet, sits an entire second population of cells, just as numerous, that for a very long time got dismissed as mere glue, simple packing material, not so unlike how the ancient Egyptian embalmers once regarded the whole brain itself, worthless, disposable, safe to remove and discard through the nose. And running through the entire system, at every single one of its countless junctions, sits the quiet, ongoing, endlessly adjustable machinery of strengthening and weakening, of long-term potentiation and long-term depression, connections growing more efficient wherever two neurons happen to fire together in useful, meaningful coincidence, connections gently fading away wherever they go unused, an entire lifetime's worth of experience, quite literally reshaping the fine physical structure of the very organ having that experience. Moment after moment, the London taxi driver's hippocampus slowly swelling with years of memorized streets, your own brain, tonight, doing some small version of the very same thing with the words you're hearing right now. This is what a thought actually is. not a spark of something separate from matter, floating apart from your body the way Descartes once needed it to. Not one continuous undivided fog, the way Golgi believed until his very last public lecture. It is this, specifically, an enormous, precisely organized population of separate, discrete physical cells, each one obeying the same simple electrical and chemical rules we walked through carefully tonight, each one shaped over your own particular lifetime by your own particular experience, working together, right now, in such perfect and immediate coordination that it simply, seamlessly, feels like being you, lying quietly in the dark, listening to a voice explain, slowly, exactly how that feeling comes to exist at all. So, here's where we leave it, back where we started, with you, lying there, in your own body, in your own room, exactly as you were at the very beginning of tonight. That same 86 billion cells are still at it, still trading their small electrical pulses back and forth, thousands of times a second, quieter now perhaps than they were an hour or two ago, easing gradually, the way they do every single night of your life, toward the slower, deeper rhythms of actual sleep. Whatever you've kept of tonight's explanation, and whatever has already slipped away into the dark somewhere between one sentence and the next, is, right now, in this very moment, being handled by the very system we spent all this time describing. Your hippocampus, patient and unglamorous, quietly filing away whatever fragments seem worth keeping. Your cortex, that impossibly thin, impossibly folded sheet, settling into its own slower nighttime rhythms. Somewhere behind your closed eyes, a small gland once mistaken for the seat of an immaterial soul is doing its actual, real, considerably humbler job, easing you, cell by cell, further from waking and further into rest. You don't have to do anything else tonight. You don't have to remember Alcmaeon, or Cajal hunched over his microscope, or the specific name of a single ion channel. The organ that heard all of it is the very same organ that will keep working on it quietly while you sleep, sorting, consolidating, deciding, on its own, in its own time, what's worth keeping. That's its job. It's been doing this job, in one form or another, for your entire life, without you ever once having to think about how. So let it do that job now. Let your own 86 billion cells settle into their slower rhythm. Rest well.